Hi, everyone. So first of all, let me, is this working? Hello? Is the clicker working? All right, awesome. Hi, everyone. Just wanted to first extend a warm welcome to everybody coming here, especially those who are traveling far distances. On behalf of the organizing committee and HCA, we would just like to thank you for being here. So a couple of housekeeping items before we begin. You should already know this is the event, so if you're in the wrong event, you still have a chance to leave right now. A quick note about media, so you can see that when you attend, we will have photography and videography and things like that. So if you're concerned about that, you should also probably leave right now. If you have any questions about registration, things like that, you can contact the organizers by that email. Of course, this event would not be possible without the generosity of our sponsors. We have fantastic sponsors who have exhibition booths next door. I think some of you have already checked that out, so thank you to 10x Genomics, MGI, and the Chan Zuckerberg Initiative, and also Scale Biosciences, who are our major sponsors. And also thank you to all the other sponsors who are also here. Please check out their booths. They're very nice booths. These are also some of the other sponsors that we have to thank. In particular, my hosting institution is HKUST, and they've also helped out a lot in this event, and the other institutions you see here. Okay, so in terms of health and safety, we have safety officers. One of the organizers, Huang, she's here, and John are the safety officers. If you need anything regarding safety, you can approach them. Name badges, please make sure that you have your name badges on. Otherwise, security might think you're not supposed to be here. So that would not be nice. The different lanyards, you can see the colors represent different roles. So if you want to approach a speaker, look for the red lanyards and you can talk to them. And again, if you have questions, feel free to email us. Code of conduct, this should be standard and common practice, but just a reminder that let's all be very inclusive and respect each other and do not harass people. I think that's standard. Okay, a quick overview on the venue. So most of the meeting agenda will be held here where you are right now. We do have a couple breakout sessions in the afternoon for both days. For those breakout sessions, some of them will be here, and there's one location which is a bit farther away, and we'll go over that when the time comes. You've already also seen there's food and beverage in the room next door where the exhibitors are, and the lunch will also be there, so go there around lunchtime, and the restrooms are just outside the hall. So again, ask any of the helpers that you see around if you cannot find something. Thank you to all the committee members who have been working on this for over a year. They're around. I'm not going to go through all the names because it might take too long, but thank you. You know who you are. In particular, I want to thank my co-organizers, Hirofumi and Wang. Okay. Also, if this is your first time at an HCA meeting, you should know that anyone can contribute to HCA. There are different biological networks for different tissues that people study. So if you're interested, you can join the biological networks. There's a link there that you can check out more information about the different biological networks. And if you're just raring and ready to join, you can scan that QR code and fill in a short form and join now. Okay, I'll just give you guys a few seconds if you want to scan that code. I see a lot of people raising their phones. That's great. Okay. Overall, the purpose of the HCA is to come together and be able to, do a lot of cool scientific discovery. The purpose of this meeting is to really update the community on what HCA, in particular, HCA Asia, has been doing in this regard. And the reason why we have the breakout sessions is really to foster collaborations between different PIs here, non-HCA and HCA. So please, participate at the breakout sessions, and we hope to be able to start many collaborations from this meeting. Okay, so a quick overview of today's schedule. We're going to have the plenary session and the keynote right after this. We would like to take a group picture, so please stay around for that before the lunch. And then in the afternoon, we have additional speakers, followed by the breakout sessions. A key thing I want to highlight is that the shuttle bus that goes back to the hotel will leave today at 7:10, so make sure you don't miss that if you are taking the shuttle back. All right, so without further ado, I think I'm going to introduce our first speakers who are going to give us a quick update on the state of the HCA. I have Aviv and Sarah on the line. Are you there? Wonderful. Hi, Aviv and Sarah. Say hi. All right, thank you.
Hi everyone. Can you hear us? Can you hear us?
Yes.
Fantastic. Thank you so much, Angela, and thank you to the entire program committee who's put so much effort into putting together this wonderful program. And we're sorry we can't be there, but we're glad to say hello and speak from afar. So it's a very exciting time in the Human Cell Atlas right now, and you'll all be aware of the spectacular publication bundle that came out. So this is a really special juncture in the project and a special time to come together. So welcome everybody who's both a newcomer and an old timer to the Human Cell Atlas. So the Cell Atlas obviously has the aim of creating a comprehensive reference atlas of the types and properties of all human cells, the fundamental units of life, both as a basis for understanding ourselves in health and also for translational purpose of diagnosing, monitoring, and treating disease. So how do we create this comprehensive reference map? Well, we want to achieve eventually for body coverage, integrating across modalities. Let me see if I can make this pointer work. Achieve high quality by working together in an interdisciplinary collaborative way to create this foundational resource. And of course, achieve diversity in both the Human Cell Atlas itself in the data, and also do that by working together with scientists across the world and across these different disciplines that I've mentioned. So the reference map in version 1 is based mainly on single cell and single nuclear RNA sequencing data. And the way we're going about building that atlas is by integrating data now, kind of this is the juncture that we're at, is basically integrating data from across many different data sets to create reference data objects. And the way we want the sort of principles by which we want to achieve this is having robustness, obviously, to technical factors when we're doing this data integration, incorporating at least all of the sort of landmark major studies with large data sets on individual tissues and organs and systems, work through the biological networks, so both the computational groups with the analysis working group, but also in collaboration with the biological networks, so that we have cell annotations with expert input from the biomedical experts, and work with annotations and depositions through the cell annotation platform, and make the data, the metadata, the code publicly available in whatever form possible. So the biological networks are as you know, they are the individual groups of PIs and their groups who are interested in particular tissues and organs and systems. And so Human Cell Atlas has grown to have 18 of these so far. We can expand that further. Right now we have 18. Each of these has network coordinators, and we're incredibly grateful to those coordinators for their leadership in these different domains. You can see the rough numbers of cells and nuclei that we have here for the different networks. So it's pretty impressive. You know, some of them have sort of many millions of cells. Some are sort of a few millions, 500,000, and it's quite incredible the number of cells that we're at to date. So around 100 million. And of course, we are also gradually gaining denser and denser spatial coverage of our tissues. but the vast amount of data is suspension cell. What we have so far are 4 version 1 data objects that we've released at data.humancellatlas.org. So the eye, the lung, the nervous system, the brain, and organoids. And what we're working on are the next wave of releases: breast, gut, immune, kidney, and liver in early next year. And then later in the year, the remaining bionetworks that you can see here. So what we're planning to have is version 1s of all of these 18 bionetworks as initial integrated data objects with initial consensus annotations. And so this is really a first draft of the human cell atlas for the suspension cell nuclear data. Right now, there are about 420 source data sets, 60 million cell profiles, FASTQs and matrices with harmonized metadata, tailored biological metadata and diversity metadata. So it's, gathering and curating this is a tremendous effort, and making sure that the metadata is in the right format. So the ingestion redesign is that the data and metadata that's contributed by the community is in the matrix store on cell by gene with matrices and that tier 1 metadata. In the data repository at data.humancellatlas.org, we've got FASTQ and tier 2 metadata, including managed access, so both open access and managed access. And then in the cell annotation platform, tier 3 cell annotation metadata, and together, that's the basis for the assembled atlases that we then provide on the HCA data portal, data.humancellatlas.org, with the both the reference objects and the source data sets. So the 3 tiers of metadata, just to be clear, include the standard technical metadata with the technologies and the technical background and how the data was generated, which helps understand batch effects, and that is provided in a uniform way across all atlases. Tier 2 is the biological information and disease metadata, which is required for downstream analysis. Some of that is in more detail is protected by managed access, and is tailored to the relevant biology of that data set by the biological network. Tier 3 is the cell annotation metadata related to cell nomenclature, and is captured mostly on a per cell basis. So in the cell annotation platform at celltype.info, there are references for all cell identities. And you can see the little animation here where you can choose the molecular data, marker genes, you can see here, that will be highlighted in the clusters, modules of genes, multiple genes, collagen shown here, and mesenchymal cell types, and so forth. In the data portal, here data.humancellatlas.org, it's an access for all the source data sets as well as these reference objects that I've just described. And you can see them here organized by the 18 bionetworks, and then highlighting the ones here. You can see the little 1 on the right-hand side. For the nervous system, there are already 2 releases, sort of in terms of the integrated data objects as reference data sets. And what's also launched, yay, want to highlight, this is a lot of work over many years, is the Human Cell Atlas Managed Access Service, which serves the raw data, after sort of registration application and review by a data access committee. And this is basically facilitated by the HCA Stichting, which is the not-for-profit incorporated in the Netherlands to facilitate data sharing under the General Data Protection Regulation. So our mission is to create a comprehensive reference map. And from here on, I will hand over to Aviv to take it forwards.
Great. Thanks, Sarah. I think it's really exciting to see how much progress was already made with the first reference map. It means that in a year or so, we will have our single cell atlas completed, but it doesn't mean that the map will be complete because it is not yet comprehensive. And in order to be comprehensive, we have to go well beyond single cell RNA-seq profiles per se. And Sarah, if you can click a couple of times into the multi-omics, the spatial levels, and capture human diversity more fully. And amongst these many different kinds of layers, HCA is especially focused on an all-out effort on the spatial level. Click, please. On the spatial level, and that's with technologies that operate across multiple scales in the human body, from subcellular to histology to anatomy. Next slide. So in order to build this comprehensive spatial layer of the atlas, we are following 3 different threads or tracks. The first track is focused on the lab technologies themselves. The second track focuses on the algorithms and pipelines that we need in order to guide the sampling, do the data collection and analysis, and both developing these algorithms and methods, but also applying them for data integration and biological queries. And then the third track is focused on data collection, from defining things like the marker panels for signature-based methods, obtaining the samples, doing the experiments, and then integrating them with other views that might not be molecular, like the histological views. Click, please. These threads are actually supported by different working groups, the Standards and Technology Working Group, the Analysis Working Group, and the Biological Networks. And you can see the growth of the spatial atlas that is happening really rapidly, which is apparent in the HCA publication bundle that just came out a couple of weeks ago, and where multiple papers reported spatial atlasing, and that included the GI tracts, the thymus, the placenta, the embryonic skin, the skeleton, the skeleton and the limbs, and the endometrium. Next slide, please. Now, just like the collection of data progresses rapidly spatially, also the computational methods. So there's rapid progress of the computational tools for the spatial domain. So here are 2 just of many illustrative examples. The Web Atlas pipeline helps integrating spatial and single cell data. And ELD is a new method and stands for effortless detection of landmarks, and that allows histology registration. Next slide, please. Now, one of the most exciting areas of development is moving these computational efforts from post-hoc analysis after data was collected and integration into more generative models, especially with AI, that would allow us to learn models from the data and then use the model in order to generate new data, or in order to be a common coordinate framework for many different kinds of data, even from other modalities. Now, what would happen if these models are successful? Then, for example, if you have already a model in hand, you can imagine a day where you take a new sample from a new individual, and Sarah, click, please, and you have only the, for some reason it didn't work. You have only the histology, and then you generate the AI of the cell profiles or virtual stain. And so 2 recent examples around these kinds of models are CAST and SHAF. CAST is from Huang's lab, and it uses single-cell spatial histology and anatomy data in order to learn a reference map. And once this map is learned, you can take any sample with these modalities from which the model was learned, or even other modalities, and you map it to the same reference. And then once it's mapped, you essentially know where it came from. So imagine, for example, that you had a little brain biopsy and you just decided to do just one kind of experiment, single nucleus RNA-seq, so you had nuclei profiles. After you use CAST, you would be able to also know the histological, anatomical, and spatial location of those cells without having to measure them. On the right-hand side, you have SHAF, which is another model from Charles Comiter and colleagues, and that's a generative model. It's trained on matching histology and genomics data, like single-cell RNA-seq, but once you train it, you can hand it a histology image, and it would generate single-cell profiles and even put them in a spatially resolved way, and it would even do it for something very idiosyncratic like a tumor. So in this way, the generated atlas is not just a description of the data that we measured, but it's also a comprehensive model of data that could exist given what we already know from what we measured. Okay, next slide, please. So now, once you have a map, you want to use it to understand biology. Next slide. And this is where it becomes absolutely essential to have great tools to query the atlas. So if we want to query the atlas to gain biological understanding, it means that we would typically come into it with a separate data set. This can be from an in vitro lab experiment that somebody did in cells or organoids or even an animal model, or it can be profiling of a sample that's not from the healthy body, but from a patient cohort that has a specific condition. So querying requires us to take these data and then ask questions with these data that we just measured, together with a reference map of the human cell atlas. And that's analogous to how you would come in, say, with a new sequence that you just sequenced, and you wanted to search it against the genomes that were sequenced in the past, except that you would do it with cells. So with reference mapping, you can annotate cells. You can ask how the composition of and states of the cells in your study relate to the atlas, and you can ask how they may be organized in space and more queries like this. So in the next slide, we can see that there's a growing number of tools from the HCA labs that enable doing this. And again, these are just a few illustrative examples. So for example, CellHint automatically harmonizes and integrates cell types when you bring in your new sample. Covet and Niche Compass characterize and identify cell niches in your spatial data. And Multimil helps query multimodal single cell data using multiple instance learning. Next slide, please. So now that the data in the human cell atlas have grown bigger and bigger and bigger and bigger, and also given the advances that happen in machine learning and AI, foundation models can help us move from these individual atlases that you might develop for each organ and system separately, like in version 1, into unified models of the entire human body. And in fact, the human cell atlas is one of the main and only data sources in biology right now, together with DNA sequences and protein structures and histology stains, all of which have existed for decades or even more than 100 years, compared to data like the human cell atlas data. And these kinds of data are the ones that are really fueling the work in foundation models for biology. So our community and members, you could say, were very prescient to know a decade ago that machine learning will evolve in one way or another and will be hungry for our data. And these models are evolving very rapidly now. So if you go to the next slide, this is a slide actually from the Human Cell Atlas meeting of last year, of 2023, and we talked then about the first generation of models. They worked at the scales of tens of millions of cells, and they were already tackling many of the classical tasks like annotation and gene program discovery, but also giving some signs of new tasks like predicting the outcomes of perturbations and searching for cell profiles. And if you go to the next slide, in the past year, we're seeing a series of really exciting developments. So there's models that scale now to hundreds of millions of cells and operate across species, like the universal cell embedding, or the UCE. And we also have multimodal LLM-like models, including Nicheformer for single cells and spatial data combined together. And there's also vigorous work on benchmarks. For example, SCEval is a benchmarking scheme to understand what complexity of data we actually, what complexity of models we actually need in order to achieve models that perform well on different tasks, from annotation to prediction, including prediction of the outcomes of perturbations or of cell types that we haven't observed in the data. And as a result of the scale of the data and the initial successes, that has really recently inspired a very large community, nicely overlapping with the Human Cell Atlas community, to propose that we can start imagining virtual cell models that would also go into the inner mechanistic workings of cells and tissues. And that's an overall really exciting direction, but many questions remain open. like what might be the true performance gains that we would get from models like these. Do scaling properties work? What do we really gain from having more data? And what are really the right, difficult, and useful metrics and use cases that allow us to test these models? If you go to the next slide, at the same time that foundation models are solidifying, we're beginning to see the next generation of approaches emerge to help us make sense and navigate these massive atlases. And these include autonomous agents that can not only engage like a chatbot through human language, but they can also plan and take actions and access and deploy analysis tools. So for example, an agent can be handed a raw, unannotated single-cell RNA-seq dataset and a question from a user, like the question on the top, what are the cell types in this dataset? And then in order to provide the answer, the agent would figure out autonomously the analysis tools to deploy to annotate cells, and it would spin out those tools, it would execute the task, it would collect the information, and it would deliver the answer back to the user, and the user can ask them more questions. And so you should watch this space. There's a few early preprints already in this area, and a lot more is likely to be coming. Next slide, please. Okay. So all of these bring us to the last component of our mission. We covered the reference, we covered comprehensive. Now we want to use this map for diagnosing, monitoring, and treating in disease. So if you go to the next slide, please. We increasingly already see the reference atlas playing a crucial role in disease studies. And this includes figuring out very basic disease mechanisms, like connecting GWAS genes to the cells and processes in which they act. There's also work in therapeutics discovery, including using some of these techniques as we screen for therapeutics, as well as regenerative medicines, where we really want to make cells with particular properties in order to supply them in the patient's body. Next slide, please. So for example, recent analyses in Alzheimer's disease relied on large-scale single-cell and single-nucleus RNA-seq atlases to tie in specific cell and gene programs and give you these nice causal chains from beta amyloids to tau tangles to cognitive dementia, which is really the clinical phenotype in the disease. People also proposed foundation models that would predict genetic manipulations that would give you cell therapies that would have desired cell states. And in IBD, there's now even clinical trials with targets that were chosen based on single-cell atlas data. Next slide, please. So these kinds of successes in diseases also open unique opportunities to use the healthy atlas for diseases that have a particularly high global burden, but are neglected or less investigated by scientists. Next slide. So there's certain diseases that are like that. They would have unique risk profiles, for example, in specific geographies, and those geographies might be underserved, or in specific demographics that are underserved. So think about conditions that afflict many women of childbearing age, like endometriosis, or oral cancers that are caused by tobacco chewing and are especially prevalent in India, or tuberculosis and other infectious diseases that are major killers worldwide, but do not get the same level of investigation and attention, say, that Alzheimer's disease does today. And so this effort is essential for Human Cell Atlas in order to be true to our equity values and our commitment to humanity, and also for HCA to successfully engage on a global scale. Next slide, please. And so let me summarize a little bit what's next. One more slide. Thanks. So as Sarah showed you, we're on track to complete our first version of the atlas based on single-cell RNA profiles. We're improving the use of the atlas through algorithms and platforms and portals, and we're building it as a foundation model of cell biology. We're also now fully building the comprehensive reference as a spatial 3D map, which is a major focus for Human Cell Atlas going forward. And we ensure that the atlas is globally representative, especially by including it as a reference for diseases with healthy global burden. If you go to the next slide, I want to emphasize that none of this has been done or will be done by one lab or one institute or even one country. It's done by all of us across 103 countries, almost 1,935 institutes, and more than 3,700 members from all over the world, including you. And so our numbers have actually grown so much, and our members have done so much that led to 448 papers up to this moment and growing. And those include the 46 papers that are in our Nature Family Publication Bundle. They were just published a couple of weeks ago, and they span atlas integration, development. I think you have to click atlas integration, development methods, and ethics and equity. And I really encourage you not just to read the papers themselves, but also peruse all the amazing materials that Nature prepared. There's front matter with reviews. There's a portal where you can go and see all the papers and images and infographics and so on. And we're really excited by this bundle. On the next slide, I want to remind you that our community also includes many, many funders. They include the Chan Zuckerberg Initiative, the NIH, the Klarman Family Foundation, the Wellcome Trust, the ERC, the Helmsley Charitable Trust, and many, many more. And also we have partners in many consortia with related areas of focus, HubMap, the Brain Initiative, Phantom, and many disease consortia from cancer to IBD to kidney disease. And I'm really happy to highlight one particular icon, the most recent one on this slide, that HCA in South Korea, which is also contributing to the atlas as well. Next slide, please. More generally, we also added, most recently, 2 industry partnership programs. One of them is for pharma and biotech to ensure that we're fulfilling our goal to impact translation to medicines. And this initiative will also help support our meetings and capacity building workshops. And the second is a new commercial discount program that we have with key vendors to help the members stretch their resources to the absolutely max, especially in data generation. They include some very long-term partners like 10x Genomics and some offerings that have to do with very new technologies like Parse and Scale, MGI, BioScrib, Singleton, VizGene, and BioLegend. Next slide, please. All of this is done by amazing people who are in this meeting. They include organizing committee members, leads and members of our expert working groups for analysis, ethics, equity, technology, and the data ecosystem, the dozens and hundreds of members of the 18 biological networks, which are organized by the organs and the systems, as well as of the regional networks, especially the Asian network, which was our first regional network, and is a very prominent one. And of course, we're really grateful to our amazing executive office team, and they make this massive global consortium run every day. Aya, Yoshi, Esther, Anya, Anne, Bridget, John, Lucia, Jenny, Christine, Ellen, and Sam. Thank you all very, very much. On the next slide, you can see some of the amazing things that they do. They're here for us in so many ways. They do program management for data integration and atlas assembly. They coordinate data curation by the bionetworks and the data ecosystem operations. They're helping handle the legal affairs and all the regulatory compliance, especially for GDPR. They provide the administrative support for the consortium. They coordinate all the scientific networks and partnerships with other consortia. They organize the meetings, like the one you're at right now, as also all the training workshops. And they manage all the internal and external communications. So not only do I want to thank the team, but I also wanted you to take that in so that you know that you can always reach out to them for these many questions and engagements. And so all of us are really united in the wish to have this global initiative. And I think if Sarah clicks one more click or two, a little movie is going to run. I think you have to run it again. Yes, it's running. To really, we want to benefit all humanity, anywhere and everywhere. And we do it one workshop, meeting, training, experiment, analysis, algorithm, annotation, project at a time. And so thank you very, very much. I wish everyone a great HCA Asia meeting.
Thank you, Sarah and Aviv. So in the interest of time, I think we'll just take 2 questions. First one is from the virtual audience, and then we can take one from the room. The virtual audience asks, are the current foundation models primarily constrained by limitations in computing or still hindered by advancements in biotechnology profiling techniques? Which one of these is more of a challenge?
I think these are apples and oranges. I think there are some limitations on access to computational resources, and we hope that this actually alleviates with time, especially as institutions catch up a little bit to where the need is. But I believe most of the challenges today are in actually the data front still. And there's two aspects to them, which we highlighted in this talk. The first one is that we have, I think, quite robust foundation models for single-cell, single-nucleus profiles. And I would say that both at the level of healthy tissues and disease tissues. If you count disease as well, there's hundreds of millions of profiles right now, and they do seem to capture quite well the cellular profiles of the body. There are exceptions with rare cells in rare locations that I think over time will be caught up. But if you look at other modalities, if you look at different single-cell modalities that are not RNA, or you look at spatial organization, we still have work that needs to be done, as well as if we want to consider questions that have to do with the impact of genetic variants on our cellular profiles and have to do with human diversity and ancestry. So in those areas, I believe there is still a fair amount of work to do, but what is exciting about these kinds of models is that they can help us identify when we've actually collected enough, and we shouldn't be diverting our resources to more collection in one area because the model is confident enough. We can focus on other areas where the model is not as confident.
All right, thank you. We can take one question from the room if there are any. Okay, if not, we'll move on to the next part of the program. So before I introduce the next speakers, I also want to just make a quick announcement about the keynotes. On the program, originally, we had 3 keynotes. Unfortunately, one of our keynote speakers is not able to be here today because of an emergency. So we are moving Jean Yang's keynote from this afternoon to tomorrow morning. So this is just a change to let everyone be aware. Okay, so our next speakers will be Jay and Shyam. They're going to give us a quick overview of HCA Asia. So Jay and Shyam.
Good morning, everyone. Can you all hear me? Okay. It's wonderful to see all of you in this room, and I just want to thank Angela and her local committee to really organize this fantastic meeting. My name's Jay Shin, and I've been part of Human Cell Atlas for many years. But together with Piero, we started HCA Asia. And today I just want to give you a quick updates, continuing from the themes of what Aviv and Sarah talked about, and how we can really leverage the community and build a strong foundation for great science and great atlas building together. So again, just to reiterate, we're here really to create this comprehensive reference map. And I think we are at a time where we can understand, but finally move into translational applications. And I think being in Asia, and I'm speaking to many of colleagues and scientists here in the region, translation is such a key importance, especially when we talk about funding and ability to do our science. So hopefully this will be an opportunity for all of us to really galvanize our efforts to push forward and really make the atlas more impactful. So when we first started Human Cell Atlas, 2017 or so, these were guiding principles, but what really stood out for us was, hey, let's really emphasize also on community and diversity. And the very white paper that we drafted, there was a strong emphasis to really have a commitment to understand genetic diversity and geographic diversity in order that we can really reach the level of equity at every phase. And so we wanted to then talk about how can we really bring out and bring people together in the region, considering that is a home to 60% of the world's population, and there are most populous countries in Asia, and also a vast amount of diversity. And however, there's lack of central governing body to really drive our research activities together. And this is just to highlight that the genome data that are currently available, we are still lacking a lot of the resource and data from Asia. And so we still have a lot of work to do. And I think this will be a great opportunity to discuss how we can achieve that going forward. So to do this, I won't name all the names here, but few leaders in the region decided to come together and discuss how we can really bring the community together to meet the goals of equity and diversity. And together with various members in here and in the audience, really enabled a lot of these meetings to come together. So what do we do? So as an HCA Asia committee, we also have strong values of building community, as I mentioned, but also strengthen the regional research alliance, and also to showcase and highlight some of the really exciting science that are happening to a global stage, and also identifying synergies and promoting collaboration and diversifying in the Human Cell Atlas. So our committee really continuously liaises with the global HCA, and you heard Aviv and Sarah talk about the whole executive office as well as the broader biological networks. So there's a continuous interactions with them. But at the same time, we coordinate these type of annual meetings and workshops, and hopefully that will bring the community together. But what really drives a lot of the activity is key flagship projects, and these are some of the existing programs as well as potential programs that could be spearheaded by one of you. So these are just ideas in which we can really come together based on key biological questions or disease that you're really interested in. And of course, we will then enable you or work together with you to figure out how we can raise funds and awareness to really drive your research forward. And then all of this coordination will be done so that we can really reach the level of equity and diversity in the global setting. So we are really here to help and support all of you to really get to know each other and to identify what are the key areas that we can really come together and focus and really achieve great science, but at the same time achieve equity and diversity. So as I mentioned, one of our key activities is to organize and hold this type of annual workshops. So I just want to tell you that this is our now 7th. So in 2017, together with Piero and I, we organized our first Human Cell Atlas meeting in Japan, Okinawa. We only had 35 people in that room. And look at so after 7 years, this is amazing. And we're just really grateful for all your participation and interest to coming to this meeting. And we have traveled around the world, and we are very grateful for all the local leaders who have coordinated these meetings. And there's still room left, so if any one of you are interested in coordinating next year, please do reach out. And really just want to thank Angela and her team to organize this Human Cell Atlas meeting in Hong Kong. And now there are about 362 in person and 109 virtual. So again, from 35 to 362, this is a dramatic increase. And once again, thank you for your interest. So I won't go into this in much detail since Angela more or less covered it, but just want to really mention that there are very exciting scientific talks as well as consortium discussions. So we try to balance this as much as we can because we want to excite you with excellent and fun science. At the same time, we are still a consortium. We want to come together and discuss how we can bring our sciences and data together. So there will be a series of new technologies and computational analysis, but also breakout discussions regarding new spatial biology technology, data management, diversity, and disease. This is something that we really want to introduce in the next phase of Human Cell Atlas. On day 2, we also have very exciting scientific talks regarding neuroscience and neurobiology and aging, as well as degeneration, and cancer, immunology, and infectious disease. But in between, we will also have breakout discussions regarding regarding, again, cancer and infectious disease, single-cell QTLs, and skin and microbiome. So these are some of these these breakout discussions are actually one of the ideas for us to really start new projects, start new flagship projects. So if any of these topics or different topics that you're interested in, please do attend and reach out to us. and we'll be happy to discuss how we can create these flagship projects. So speaking of flagship projects, one of the ideas that came out in one of the meetings we had in Korea in 2018, Shyam, Prabhakar, and Woon-Yang Park and myself said, oh, let's come together and start a project. Thanks to funds from CZI, we were able to start this Asian Immune Diversity Atlas. I just want to say that it's been a tremendous opportunity to collaborate across various scientists around Asia. And in fact, after these many, many years and hard work have led to very successful and fruition of publication that were either accepted or published in Cell and Cell Genomics and Nature Genetics. So all of these efforts started from this type of very room in 2018, where various people got together and say, hey, let's start a project together. And after several years, we now have achieved a very great milestone in terms of demonstrating how we can reach the level of diversity in terms of immune repertoire, but also find new discoveries in genetics. So hopefully it will encourage you and motivate you to talk to each other and think about new flagship ideas that can be very successful. So with that, as I mentioned, the last point, and ultimately the goal is to reach equity and diversity. And for that reason, I want to bring Shyam to the podium and have him discuss and share with you some of the task force activities to achieve this goal. So thank you.
Thank you, Jay. Real pleasure to be here and building off the state of the HCA and Jay's introduction to HCA Asia. Okay, just a minute. Okay. I want to present the HCA Diversity Task Force recommendations here, give a broad overview. Lead up to that, really wanted to give a shout out to CZI for funding a whole bunch of projects all over the world that help to characterize and describe human diversity, right? And as you can imagine, this is extremely important. It's a hugely important endeavor, and in many databases, including GWAS databases, GTEx and other like that, despite best efforts, there is still not enough diversity in all of the data. And as you can imagine, that limited diversity limits our science and limits our ability to transform or translate that science into clinical benefit, right? So the HCA has really been a pioneer in redressing that issue. And I think over this coming year and the next year, you'll see all of the data from this massive wave of studies, and that'll help to transform our understanding of what it means to be human. Now, the HCA Diversity Task Force was given the charge of providing some guidance to all of these exciting scientific efforts all over the world in the HCA, so that new people coming in could get a sense of what we hope to achieve in the HCA. So above all, ethically and scientifically appropriate processes for conducting such research, and then what metadata should we use when we study human diversity, because that's important. If each group uses its own metadata spec, then it's hard to compare across studies in the HCA, and that goes against the whole philosophy of the HCA. And then metrics and resolutions. If we say we want to achieve something, then we want to have some numbers. Those numbers may not be definitive, okay? Every measure has its flaws, but some numbers to be able to track how well we're doing in the space. And finally, some targets, meaning after 5 years we want to be here, and after 10 years we want to be here. Qualitative targets as well as numerical targets. And ambitious goal here, I'll broadly describe, and then we'll put out the recommendations in public soon. The idea is to maximize benefit while minimizing risk. And then, of course, consult with the local communities when you do this research to understand their sensitivities and their goals, and build capacity. And, of course, do that in a way that's consistent not only with your principles, but with their ethical principles in their community. And then focus also on the underrepresented communities, marginalized communities, and benefit sharing with them, right? So those are the broad principles. And so we've, very proud to say we've laid out the metadata spec now, and it will hopefully be ratified soon. And we want to cover all of these aspects of human diversity. There are more, but these are the major ones that the committee identified, which is diversity in genetic ancestry, self-reported ethnicity, and ethnicity includes an element of culture as well. Language, diversity in geography, age, sex, gender identity, and of course, environmental and lifestyle factors which have a major impact on our health, right? So human diversity metadata are important for every single bionetwork, and every bionetwork will have its own specific diversity data that they need to collect. But we try to build a baseline for all of them that's shared across all bionetworks. Okay? And that'll be part of the HCA Tier 2 metadata, which will not be, for privacy reasons, it will not be publicly accessible, but still important for our research, right? It'll be controlled access. So I mentioned all of these major kinds of metadata we're collecting, and hopefully all of you, when you start a new study, you will access this metadata spec and make that part of your donor engagement, okay? So what are our metrics for human diversity? Very broadly, right, we want to measure geographical diversity of the samples in Human Cell Atlas, which means number of samples per country, per continent, and also some measure of geographical distribution of the samples, though of course the finer you get in terms of geographical location, the more your privacy concerns arise. So that's really something we want to assure the whole community. We're not trying to collect information that could compromise the ethics or safety of the participants in your study, right? It's finally the prerogative of every single researcher to collect and report only the metadata that they consider safe to report. And of course, proportion of countries in each continent represented, we'll measure that as well. Self-reported ethnicity, and here are some examples. Then genetic ancestry, we're not trying to bin genetic ancestry, more that we'll create a map in some kind of PCA space of the ancestry of all our samples to see how diverse, how broad our collection is. And then finally, our goals, this is the hard part, we decided to stick our neck out and specify some concrete goals at the 5-year mark, at the 10-year mark. Of course, we want sex balance. That's relatively easy, but then we also have numerical goals for how many countries, how many continents at 5 years and 10 years for how many bionetworks in the HCA, and also environmental diversity, some basics of urban versus rural environments, okay? And then for all HCA atlases, we also recommend that your version 2 of your atlas should be more diverse than version 1 of the atlas. Why is that? Because we're starting out from a baseline where many parts of the world started doing large-scale single-cell research a bit later, right? So that means we're starting out from a place where the diversity in our database is not where we want it to be. So definitely to catch up, we want to make sure that version 2 is better than version 1. This sounds trivial, but it's not, okay? Without naming names, for other consortia doing other kinds of large-scale genomics, their diversity has been, level of diversity has not improved in 10 years, right? So unless you make a strong, explicit commitment to going version by version improvements, we run the risk of facing the same situation 10 years later, and that's not what we want. And then a major recommendation from the diversity task force was that the HCA expand its disease efforts. As you know, the HCA has already made a major contribution to COVID studies during the pandemic, putting out an atlas of healthy and COVID states. The idea is, and that's part of the HCA philosophy, you build a healthy reference atlas in order to better understand cell states and cell processes in disease, right? So now it's about time in the maturation of the HCA for us to take all the atlases that are coming out, and you'll hear some of it from Chloe and others today. It's time to start translating the benefit of those healthy atlases by using them to understand disease states. So this is an opportunity for everybody in this room for sure, and everybody in the community, to collectively define which diseases we're going to work on together across the globe for local benefit, for global benefit, looking at the burden of disease, feasibility, relevance of single-cell profiling, and so on. And we believe that disease applications are essential for achieving the equity and diversity goals of the HCA, because disease is forefront among the concerns of national funding agencies in many parts of the world. So for geographical diversity, these are our numerical goals. The first part is our 5-year goals. The second part is our 10-year goals. And by 10 years, we want to hit at least 50 countries across a number of bionetworks, right? And in 5 years, we want to hit this number. So blood is the easiest. We want to hit 50 countries for blood in 10 years. And then skin and nasal swabs, perhaps the next most easy. And then at least 3 other bionetworks, we want to cross 10 countries in 5 years, right? When we say country, we mean we want to have a whole cohort, not just one sample from that country. So these may seem ambitious, but eminently doable, we believe, based on a survey of the progress and the trajectory of the Human Cell Atlas so far, right? And so going a bit more into disease applications, I guess the key point is that many countries cannot fully participate in the HCA based on their local funding prerogatives if you don't consider disease, right? And so if this is the right time, not from a diversity perspective alone, but also from a benefit perspective for the HCA to expand to that. And then these are the key criteria. I put global and local disease burden on top, but it's not the only criterion. There may be some diseases like, say, alcoholism, where it's harder to access the biologically relevant disease tissue or the causal disease tissue, which could be in the brain, for example. Or there could be some environmental risk factors where only very specific tissues can be accessed that are relevant for studying the effects of that. So we really want to pair the disease burden with the relevance of the tissue, complementarity with existing consortia, and the expertise of the existing HCA network, right? So I encourage all of you to take a look at the recommendation of the Diversity Task Force. There's one poster that Huang will be presenting to give an overview of that. And also, please take a look at this URL. Hope to have your feedback and questions about this. We put out our recommendations, about to put them out. We want to hear your voice to see if they're appropriate and how you can engage. Thanks very much. Do we have time for questions or just move on? Okay, maybe time for one question. Okay, let's move on then to our opening keynote. Great pleasure to announce Alexandra-Chloe Villani, who's going to kick off the session. She's an investigator at Mass Gen, and an assistant professor at Harvard Medical School as well, and also in the Broad Institute. Chloe got her bachelor's degree and PhD from McGill University, and then moved on to do a postdoc in Aviv Regev's lab at the Broad Institute. During a PhD, she studied IBD susceptibility genes as well as a new Crohn's disease susceptibility gene. And then in her postdoc, she became famous for single-cell profiling of immune cells. She's really been a pioneer in this area. Her research has been published in many journals, including in Nature and in the recent HCA publication package, Science, Nature Genetics, Nature Immunity. She's got many awards, including the Janeway Award, Louise Berlingay Postdoctoral Fellowship, Banting Postdoctoral Fellowship. And she's really a pioneer and world leader in large-scale population-based and single-cell profiling techniques, which she won't talk about today, but I want to highlight that. And high-throughput functional assays for monitoring the states of immune cells in health and disease, autoimmune disease, cancer, and so on. Many of you already know her through her leadership of key aspects of the HCA, and indeed Chloe is everywhere in the HCA. Among other roles, she sits on the executive committee of the HCA, the organizing committee. She leads the data ecosystem and the cell annotation platform, and really a driving force behind some of the new initiatives you're going to hear later today in the breakout sessions. Please join me in welcoming Chloe.
Thank you so much for the very thorough introduction. And I love to, it's food for thought, Shyam, that you think that I'm everywhere. And it's such a pleasure to be here. I look forward to meeting all of you, interacting with you. I'm here for the entire meeting. And so what I thought I would do today is give you a sense of all of the tooling and procedures and governments we have built to start doing Atlas assembly. I will admit that the presentation is packed with a lot of information. The goal of this presentation is not for you to remember everything. This material will be made all available to you. This is more about making sure that you understand what we've built behind the scene to enable doing Atlas assembly. Okay, and so I put this slide that's perhaps overly simplistic in terms of summarizing our history of HCA. Which started in 2016. What I mostly want to highlight is that as a grassroots initiative, we've been evolving over the years. And like any grassroots initiative, we had to reinvent ourselves to adapt to all the progress we are making. So we started by building the communities, building expertise across technologies, developing analytical frameworks, doing data generation, but we're entering an exciting new phase, which is the phase of going from data generation, community establishment, to assembling our atlas here in 2024. And you've heard some highlights of this in Aviv and Sarah's introductions, and I want to expand a little bit on this today. And so you've heard that we're currently building atlases across the 18 different biological networks, all working together to create this first reference genome, reference atlas. And there's a lot of different single-cell efforts out there, and so I want to point out why we think the Human Cell Atlas endeavor is a little different here. We are trying to establish gold standard reference atlas. What do I mean by that? Like, how are we different from the other efforts? We are taking a consensus community-led approach to do that. I'm going to expand on this during the presentation, and applying rigorous standards to achieve so. We are operating at an unprecedented levels. You've heard in the introductory remarks that we now have over 400 different data sets, and we're aiming for global representations. You've heard about our version 1. I want to highlight that this is not the end. This is the very beginning. It's an exciting time, right? As our community grows, as we generate more data, we anticipate iterating this atlas. So what do we need to iterate with this atlas? It means we need to think about centralizing some of our governance, which I'm going to go over, and we need to start formalizing some of our infrastructure. And so you saw this introductory slide about how HCA is organized earlier on. One of the newer group that was added under the HCA organizing committee, which is the group of scientists around the world that provides scientific leadership for HCA, is this data ecosystem oversight group. And so let me introduce you some of the key advents of the last year, because a lot have happened behind the scene. So I'm going to go through all these different 4 advances today, including how we created this data ecosystem oversight group, how this is meant to help advance the atlas, how you can now submit your data and manage access, and how we're excited to open this up to broader contribution. And so this is a bit wordy, but it's to give you a sense of the scope and mandate of the data ecosystem oversight group. We're here to coordinate the whole ecosystem, which we're going to go over. We are responsible for data ingestion, data storage, the integration of data sets, cell type annotation, and the HCA data portal. And we're there to help and receive regular updates from the different team leads, and to provide updates and recommendations to the scientific leadership body of HCA, which is the organizing committee. And we're here to liaise with different groups. These are the different phases of it. This is not a typo that some of the names are in blue, because these individuals are actually here at the meeting, and so I invite you to all connect with me. Shyam Prabhakar is actually the co-chair of the Data Ecosystems Group, so you have representation right here in Asia, which is exciting. You also have Joseph Powell that I saw earlier today, representing Australia. We have Lucia Robson and John Randall, members of the executive office that are also members of our Data Ecosystem Oversight Group that can help liaise between the different groups involved. And so I'm summarizing through this cartoon our different responsibilities of governments. So as I mentioned, we oversee everything integration, managed access, and operations. And we are responsible for overseeing some of the policies being developed to help manage the data ecosystem infrastructure that I'm going to tell you more about. That includes the matrix store, the cell annotation platform, the data repository, and the data portal. And with all of this, we liaise with the different working groups to make sure that the work is well coordinated. And so I want to tell you now a little bit about some of the data ecosystem in greater detail. Some of these slides were shown in the introductory remarks from Aviv and Sarah, but I'm going to go in slightly more detail to kind of go over metadata standardization, the ingestion redesign, the portals, and the CAP. And here what I'm hoping to do is to give you a glimpse of how you can all contribute to the Human Cell Atlas, whether it's through contributing data to kind of gives you a sense of where to start, or whether it's to engage with the Atlas assembly effort if you want to engage with some of the cell annotation process or the cell annotation jamborees. And so I'm going to start with metadata standards. Our goal was to develop and implement an updated metadata schema that would involve standardizing the file format for summoning data. And we're focusing first on gene expression space. And there's a few reasons to do so. The first one is of course, as we do the 18 biological network atlas assembly to integrate all the data, we need to have the same standardized format. But the second one, you have to understand that this is only version 1. Version 2 means also connecting cells that are shared across different organ systems together through integration. And we can only do so if we all standardize the way we have compiled our data. And finally, if we are to develop some portals to do queries and software, we can only do so if we all work with the same standardized file format. That may seem obvious, but it's not so obvious in a day-to-day work when you have to go beyond just publishing your academic work. So you saw these 3 tiers, but I'm going to, through Aviv and Sarah's talk, but I'm going to go slightly slower to go over them. The first one is what we call tier 1, is about 35 different fields. These are not challenging fields to collect. We're asking you, where are these cells, where are these nuclides, what's your institution, where is your data coming from? None of these have identifiers. These are technical information that are required to do batch correction, to do data integration. And they are uniform across all 18 biological networks. Doesn't matter if you're heart or lung or kidney. These are type of information that are uniform and harmonized across all. To give you an example, I cited a few, but it can be, what was your sample ID? Was a broad demographic, no identifier, so we're not asking you for specific age. It's about age range, but we must define the age range similarly. It's about experimental details. It's about sequencing information, which platform did you use, what reference genome did you use, and so forth. And this is managed by the CZI Cell by Gene team, with whom we partner to do this, and they're our matrix store. The second tier is a bio-network specific to some extent, and it's also the work that is tied to the genetic diversity bio-network that spans all biological network. This is about once you have an integrated object, how do you do biological queries, right? What are the biological queries you want to do for your atlas? Some of these files can be managed access to protect for all of the identifiers, and as I mentioned, it can be specific to your biological network. That will include ethnicity information, but it could include very specific fields in terms of the particular organ you're studying. So I gave you as an example COPD for lung. This is managed by the HCA data repository team, and that includes our raw FASTQ files. And finally, you heard of the cell annotation platform. So in order to have a resource that is useful to the entire community, we must also agree on how we call the cells. We must agree on developing a common lexicon across all organ systems so we can do the biological queries. And I'm going to expand a bit on this later on. So it includes getting information about cell label and annotation rationale, such as the marker gene, cell ontology terms, and synonym. That's managed by the CAP team. And so you're at the very top as a contributor. And you can contribute. We encourage you to connect with your biological network coordinators because it will help coordinate the work, help align priorities in terms of data sets being integrated. And then you feed into these 3 different branches that I just mentioned. The HCA matrix store, the HCA data repository, and the cell annotation platform. And so the matrix store is currently being co-led by Jason Hilton and Brian Evermand. You have the HCA data repository with Tony Burdett, Gabi Jordanova, Idazuki, and Arsenio Chadzi Giorgio. And the cell annotation platform with Evan Bithersens, Marius Fute, David Osumi-Sutherland, and Jasmine Belfior. And we've compiled detailed instruction on how to compile these 3 different type of metadata. That I invite you to scan the QR code. You can also just go to the HCA data portal and you're going to get this information as well. You can also reach out to all of us. I'm going to give you some information about on the HCA data portal so that we can walk you step by step on how to proceed this. The 18 different biological networks are working towards our first version of their atlas and compiling this tier 1 metadata and this tier 2 metadata that I've told you about, and this is currently work in progress. So, once you have information across all 3 different tiers of metadata, you can work towards atlas assembly. For this first version of the atlas, we're grateful to CZI, who provided some initial funding to help fund a centralized team that bridge between the analysis working group of HCA as well as the cell annotation platform, as well as every individual biological network in the cell annotation platform to assemble these different atlases. And this is led by Mata Lukin and also managed by Lucia Robson at HCA Executive Office. And all of this information, once assembled, is going to be available to the HCA data portal, which is managed by Dave Rogers. And so if you go on the HCA data portal, through the HCA data portal, you'll be able to access the matrices, the end-data object, the full metadata, the link to the cell annotation platform, the link to access the FASTQ files and matrices, and access to request for managed access data. So to give you a sense, I'm showing you the page for the long. So every biological network is a little different, but similar in format. You have the overview of the biological network at the very top. On the far right, it tells you the number of atlas you would have, the numbers of disease that were involved, the total number of cells, and at the very bottom, the name of the data sets and slightly more information. And so if I click to this hyperlink, it tells me exactly the data sets that are part of this atlas. On the first column, the second column tells the species. These are all Homo sapiens. It tells you which disease and the number of cells included. This whole ecosystem is actually well coordinated through operations, meaning this is me showing you a little bit under the hood how this is done. And so every members of the 3 different pillars of the data ecosystem meet monthly under the overview of the HCA Executive Office. And the way they coordinate all of this, they have different tools to do so. But one of them, just to give you a sense of how we're doing this, we have an HCA Atlas tracker that enables the team to know which data sets you're submitting. It gives transparency to all stakeholders involved in ingestions. It gives us a point of contact between our data ingestion team and all of you. It breaks down outstanding activity tasks so we can do follow-ups with all of you. It displays deadline for ingestions and link to different source data sets. And it helps us to kind of give updates between our team and then the different platforms we need to coordinate with. So this is an example of current ongoing cell atlas ingestion tracker for the heart. So you have the different data sets being ingested in the first column and the publications. Most of them are published in the example that I'm giving you. And across the 3 different pillars of the data ecosystem, it tells you where we stand, right? So it gives you a sense, a little bit of how we are organized behind the scene. Importantly, for any data sets to be part of a version 1 of an atlas, we required that the data sets be tied to all 3 layers or all 3 pillars of the data ecosystem, which means that your data must be submitted as a gene expression matrix in the right format with a tier 1 metadata. It means that we expect you to contribute to the tier 2 metadata in terms of the biological fields decided together with you and the biological network, as well as we require to know where your raw FASTQ files are located. We also require that you contribute your cell annotation metadata to CAP, which I'm going to go through over in a few minutes, for our data sets to be part of any versions of the atlas. Overall, beyond these 3 different pillars, these are the version 1 criteria to be, for an atlas to be approved by the organizing committee, again, which is a scientific leadership body of HCA. So members of the biological network must have approved which data sets are included. It must include all landmark studies. It must be robust to technical factors. That means we must have integrated based on the tier 1 technical metadata. Annotations of the final objects pushed forward as our version 1 must have input from key representative. And as I mentioned, the raw data and the gene expression matrices must have been stored. We have provided a lot of documentations that you can access. Again, you have a QR code here on our HCA portal that includes updated policies, instructions to submit your data, and we're about to release very soon some standard operating procedures for data integration. I was fortunate enough to take part to the AIDI workshop yesterday, where we actually talked a little bit about the use of unpublished data and embargo data to HCA, so I want to talk about this very briefly. So we also encourage people to share unpublished data set. We adhere to the 4 data principles. We are committed to protect unpublished data under embargo until the atlas can be incorporated in a scientific journal. It means we still encourage all of you to publish your academic work independently, but you have an opportunity to have a second win with the same data by contributing to an atlas. And you can do so by contributing your unpublished data before you're completely finished with your first paper if you wish to do so. I'll give you an example for myself. I'm part of the Immune Bio Network. I actually help co-coordinate the Immune Bio Network. And as part of the current atlas assembly effort, I shared 3 million of my unpublished cells with the effort. And I'm still working towards publishing my own work, but at the same time helping the atlas assemblies go faster by having shared that data as well, so that can be a win-win. We have some policies that is out there in terms of the unpublished data that I invite you to read and reach out to the executive office if you have further questions. There's no one-size-fits-all about how one should do atlas assembly. When you think about it, in many cases, we need to tailor them to every organ system. So what we've done over the last year and a half is work together to develop a standard operating procedure, which we're about to release. You should hear about it in the coming months. And it's divided into 3 different broad chapters. The first one is about project setups. We give you a recommendation about how to assemble your team, how to define the scope of an atlas, how to ingest the data and metadata that I just went all over with you, how to think about assessing quality of your data, how to do batch analysis. We don't tell people, we don't tell all of you, you must do this quality filter, you must do this batch analysis. But we're asking everybody to actually do some quality filters and to tell us why you did them and how you did them. For the second phase, the second chapter is about data integration. We go over how to prepare to evaluate your integration strategies. That includes defining some forms of ground truth through cell annotation, for example. How do you go about evaluating your integration methods, whether it's through qualitative or quantitative metrics, how to perform your data integration, how to think about downstream clustering and analysis. Again, we don't tell you this is the integration method you should use, this is the metrics you should use to assess your integration. We're asking you to test more than one integration method, more than one set of parameters per integration methods, to use some metrics, some qualitative, some quantitative metrics, and to tell you which one you used and why you ended up selecting a particular integration method instead of parameters to proceed with your atlas. We ask people to be transparent, to release their code so that the work can be reproducible moving forward. And then you can proceed with any type of biological queries, including community input on annotation. And so that brings me to the next piece of my presentation. There's a lot of work to do at atlas assembly. Once you finally have the objects, it's not the end of the work, it's actually the beginning of the work in terms of the biological query, right? How do you go about agreeing as a larger community about how we call our cells? And so that brings me to the cell annotation platform. You've heard a little bit about it during Aviv and Sarah's introduction. So I listed to you the version 1 of what needs to be done for an atlas to be approved by HCA. Version 2 of HCA, I told you we're going to be iterating in the years to come. The HCA organizing committee has not finalized yet its criteria, but one criteria that is already agreed upon is that every atlas will have to go through a community consensus annotation effort. And that needs to be done on the cell annotation platform. And so I'll give you a sense of how this is getting organized behind the scene, but I'll start by discussing why I think cell annotations can often be seen as just an opinion, right? And myself included, I've spent the last decade doing a lot of manual annotation of myself. I'm an immunologist. It's like my party trick. I can look at marker genes and tell you which cell it is. But very often people ask you, well, how did you figure this out? And it's like, oh, it's all in my head. And that's not really helpful to be all in my head. I need to be able to share this information in a structured way. And it turns out that very often with my colleagues, I differ in terms of how I define the cell types. I may even differ in terms of molecular signatures and marker genes that I use. I differ in terms of the vocabulary and terminology that I use, and that's really problematic. And so what we asked a couple of years ago is how can we systematically and more objectively compile and compare cell annotation provided by different domain experts to derive a consensus? How can I take everything that's in my head when I think about my favorite monocytes and dendritic cells and structuring in a way that other people can decide if they agree or disagree with me? And that's what led to the establishment and development of the cell annotation platform, or CAP solution. It is a friendly user interface that is meant to be a community-driven platform to create, explore, and store annotation. We've built some infrastructure, and I'm going to go over some of it, to accumulate, share, and analyze annotation terms that are associated with molecular signatures so we can interpret cellular identity, and with time, gather enough information so that we can help improve some of the automated annotation with cell type prediction models and work towards developing consensus nomenclature altogether. To do so, we structured the metadata with 12 different fields. Again, you don't need to remember all of this. This is all documented. But it includes the name, the cell label set. We want to understand the rationale of why you call a cell, right? What did you do unbiased marker gene analysis? How did you define marker gene? Was it pseudobulk? Was it a decision tree? What marker, did you use legacy markers, or was it completely unbiased? Were there known cell ontology terms? If not, what is the closest term? Are there any known synonyms for your markers? Did you do your annotation manually, or did you use an algorithm? And if it was an algorithm, which one and in which version? And by compiling this information in a structured way, suddenly you have the necessary context to consistently and accurately work with annotation to construct atlases. We're set now to start comparing and contrasting annotation between different groups if we do so. And we have built a team that is there to work with you on queuing this information and uploading it on CAP. And I was delighted that a team from Prabhakar's team in the ADA effort, including Huang and Ilura Violen Buyamin, have worked already with CAP to put all of the ADA information on the cell annotation platform, and I invite all of you to get involved. The cell annotation platform, think of it as a platform that has many different modules. So you have the home page, you have a data upload and publish page. For sake of time, I won't go through all these details, but we offer private space so that you can work on your data until you're ready to publish it publicly. We have an interactive annotation user interface where you can only browse molecular signatures pathway and do live differential gene expression analysis. You can also start doing annotation transfer and de novo cell type predictions. We are offering tools to compare annotation between different data sets. And with time, as we develop consensus annotation, the same way gene cards exist, we're going to also offer cell card view that summarizes all the consensus information and annotation metadata that have been developed by the community. And so I have a few videos. I was told I don't have a pointer. And so I'll do my best to describe what you see. I wanted to make sure that you kind of get a glimpse of how it works as opposed to just doing a dry description through a slide. So this is an example of landing page. I can go and click on the data sets, and it's going to list you different data sets that are already there. If I click on the data sets, it gives you information about this data set as well as all the cells and the number of cells per subtypes that are in there. If I click specifically on the data set, now I'm going to scroll and show you color-coded all the different cells and number of cells in that particular data set. By the main page, I can click on cell relationship, and it's going to tell you the relationship between these cells and other data sets of the same class that I'm showing you. And this is, I think I mentioned, is a cell long data set. And at the very top, my option is to kind of go and click on molecular data. It's going to take a moment to load because what you have to know with the cell annotation platform is that this is live computing. It's not all pre-computed, which means that you can actually interact with the data sets. So now you have the embedding that has been uploaded with the different cells that were defined. Here we have the different marker genes, color-coded at the very top by the different cell type, and the brighter the color, the more specific a marker is. And I'm scrolling down to show you all the different marker genes that were defined by their data contributors in that data set. You can zoom out. And then I can click on the particular markers, or you can look for that particular marker by pinning it. It's going to show you where PPAR gamma, for example, is expressed. It's expressed in the alveolar macrophage. If I hover over at the very bottom of the screen, you can see it tells you the levels of the gene expression that is detected. You can go back to the whole set of the data set. I can go to the author cell type here and look at the full list of data set. What you could do if you had not annotated that particular data set, let's see if this video works, I can actually have selection tool. I can zoom in, and I can select these cells, and I can try to figure out where will be the marker genes that are specific for this cluster. So I'm selecting these cells. And now at the very top, I can go and do differential gene expression. I can click differential gene expression. I can compare to the background or to a custom reference. And now it's going to compute live. The markers are going to be specific for the cells that are highlighted that I'm showing you at the very top, including some of these top marker genes. I can click on the marker genes. I can hover over. It's going to tell you a summary of what the biology tie to this particular gene. And I can pin a gene. I can select a gene. It's going to show you where exactly it's expressed. In this particular case, you can see that it's expressed on both clusters that were there, which suggests that, and we're simplifying, of course, this is a single cell type. And I'm showing you the marker genes here that are going to be specific to that cluster in red compared to all other cells that are in blue, saying that these genes are particularly specific. So I may be ready to create a label set. I'm going to give it a name. This is a cell type, just for sake of example. And now I'm going to have, I can create add a label for this group of cells. And so I'm selecting it, and now I'm going to start entering all the metadata that I told you about for this particular cell type. So the names, the synonym, and you can see that there's prompt that are appearing. This is to nudge the user to go towards more consensus labeling. And so for sake of time, I'm not going to let the whole video play, but you can see you're entering all this information. Now, if you've done all the annotation outside of CAP, we also have file format where you can complete this and upload this on CAP. When you're done, you can do submit, and now that cell is labeled. So let's say we all do our homeworks. I hope we all do our homeworks. We compile this information. Now we have it in a structured way, which means that we're set to compare and contrast annotation. We can start thinking about how to do cell annotation jamborees. We can compare our opinions in a more structured way. We can do rounds of voting and address our disagreement. And so what could happen? In the best case scenario, we could agree and there's nothing else we can do, but we still want to compile who agrees, who doesn't agree. And so what we've done over the last years is we've implemented feedback feature. So here I'm showing you an example from the gut cell atlas. And so on the far left, you have the different cell types. And so let's see, this is going to play. Yes. So I'm going to go and select a particular cell type. I'm going to select the enteroendocrine cells. These are some of the marker genes that were defined. It turns out that I want to select these cells. At the bottom, you can say provide feedback, bottom right. And I'm going to say, you know what, I agree. And then you can leave a comment or not leave a comment and send your feedback. And we still want people to tell us if they agree because we want to compare the statistics about people who agree or disagree. The other option that may happen is that you actually agree with the molecular signatures. You agree that a group of cells share molecular identity, but you actually disagree with how we labeled it, or you agree with the metadata we use to justify that this is a unique cell type. You disagree with the rationale. So for sake of time, I'm not going to run the video, but what you could do is you can have the whole metadata summary come up, and at the very top, you could click Refine Metadata, and then you're going to have the option to tell us where you disagreed and provide alternative solution. You must provide alternative rationale if you disagree with the annotation metadata. Finally, you could actually disagree that a group of cells should not be together. You may tell us, actually, 2 clusters should merge, 2 clusters should split. So I'm going to give you an example here with the T cells. So I'm selecting the T cells here, and I'm showing you some of the marker genes evidence. And this is as a simple example with the T cells. We're like, you know what, this is way more complex. There should be more subsets. So I'm going to select the cells. And at the very bottom, I'm going to go and provide feedback. I'm going to go and I say, I disagree. And I'm going to say, you know what, I want to split the cells. And here you need to tell us how many groups it should be. And for every group, you need to give us what would be the marker genes so that we can start compiling that rationale. And so for sake of example here, we're talking about mature and immature cells. And you can, again, you get some prompt in terms of the information you can add here. And at the very end, we ask you to provide a rationale for why you decided to do so, right? So at the very end, you're going to see entering some explanation. And then when you're done, you can submit your feedback. So for sake of time, I'm gonna move forward. At the end, when you've done this across all integrated atlases, we can provide feedback summary for every cell subset that I'm showing you here. With the green arrow up, people that agree, and arrows down, people that disagree, with the comments of why they've disagreed. And for sake of time, I'm not going to show the videos, but they're going to be all available for you on the CAP website, on YouTube. You can see how we can project the feedback summary on every clustering embedding, so you can see the areas where you see broad agreement, broad disagreement. Importantly, this is a transparent process. You must register. It's free to track who provided the information. And all cell annotation authors and reviews will be made transparent and public. The resource itself is publicly available and free. And this is a HCA-centered product. And the goal, again, is to provide a framework to develop this consensus annotation for reference atlases. It's important because by doing so, we can work towards consensus. It may turn out that there's 20 different groups that identified the same groups of cells as a unique identity, but we gave it 20 different names. It's actually helpful to know that we have 20 different synonyms, and then the community can decide together what should be the official name. There's a lot of amazing automated annotation frameworks out there, but at the end of the day, you still need a detailed reference for these automated annotation frameworks to work very accurately. So here, by compiling detailed annotation metadata, we have an opportunity to really improve the future iterations of these automated annotation framework. There's a lot of efforts leveraging cell ontology to do integration, but a lot of the cell ontology terms are inaccurate or not existent. So we have an opportunity to update these cell ontologies and improve future iteration of cell atlasing. And as importantly, we can identify where as a community we tend to disagree in terms of cellular identity, and that can help us focus our next efforts in terms of data generation and iteration. It may just be that we need more different type of modalities, greater data sets together, because at the end of the day, a cell identity is not just defined by its RNA profile or protein profile. There are so many different layers of identity that are critical in defining a cell subset. And so there's detailed information on how to provide information onto CAP. It is part of the instruction for submission for the data. We have a user interface ready to go and a team ready to support you to do so. And so it's the same QR code that I show over and over again. You can also get this information on our HCA website. And this is Evan Petersens and Mary Fute are some of the leads of the cell annotation platform. They also want to hear from all of you. I've shown you some examples of user interface and features, but actually we are working with every biological network so we can tailor these features for cell annotation. Only a few slides left. I've mentioned managed access. We're very excited about releasing this. And so why is it so important to us? We want to make sure that scientists around the world can contribute data sets, right? We want to empower every bio network to create a more thorough and diverse atlas. Jay and Shyam tackled this, right? It's a key priority for HCA to have a diverse atlas. And so we needed to develop a platform so that everybody could contribute their data. So we provide end users access to rich metadata and raw data from virtually anywhere in the world. HCA can store data requiring access, such as FASTQ files and personal information and ancestry, and is supported by International HCA-specific data access committee. How do you access this? You saw this briefly in the introductory remarks. You can visit the HCA data repository at data.humansatlas.org. You can search for data sets you'd like to access. And then you can click on, see the tag access required, and then you click on request access and you fill out the form. And so we were very proud that ADA was the first data set to be submitted for this. And so to conclude, we really want to expand project participation you heard about these 18 biological network. If you go on the website, you're going to see the different coordinators, but the coordinators are there to, as the word says, coordinate. They're here to help to play matchmaking, to try to see who has interesting data sets. How can you contribute? But more importantly, what we need is champion across different bio networks. So for example, I represent immune. If you have immune cell data set you're excited about, you want to contribute to it, please do reach out. Please come and find me while I'm here. We want to expand project leadership geographically, and that's why I was so excited to come and meet all of you here, so we can, expand the next generation of our atlas. And at the end of the day, you are all a key to expanding our next generation of atlases. The biologists, the bio-network are really driving it. There's a lot more that needs to be accomplished. So to give you a sense of where we stand right now in short term, we're trying to finalize compiling all the tier 1 metadata for all the data sets being currently integrated. We are working to finalize these biological tier 2 metadata across every biological network to finalize a version 1 of the atlas. Medium terms, you're going to hear from us. We hope you will all engage in the Cell Annotation Jamboree in 2025 and 2026. Importantly, you don't need to have contributed a data set to the version 1 of the atlas to add your voice to the Cell Annotation Jamboree. We actually want to hear from all of you so that the product that emerged from all of this is something that you feel your voice contributed to. Please ask for support from the HCA Executive Office, the KEP team, the integration team, the data repository team. If you have interesting data you'd like to contribute to, you can also email us at ranger-team@data.humancellclass.org. It's a team sport. We need all of you to engage to make HCA successful. I'm so excited to see so many people attending this meeting, and I look forward to meeting a lot of you during the rest of the meeting. And I'll be happy to take some questions. Thank you.
All right, Hayek.
Can you tell us the difference between depositing the data to HCA versus like a European, the EGA archive? Do you expect to have like a role for HCA only, or do we need to deposit our like raw data in the EGA and HCA through a different formats?
Thank you so much for your question. And before answering that question in detail, I want to highlight that we have a breakout session this afternoon that I'm co-chairing with John Wendell, where we want to have this discussion with all of you about data management and data access. So first, in terms of the matrices, so not the raw data, but the gene expression matrix, or with the technical metadata, we do ask every contributors that want to contribute to HCA to submit through a framework, because we ask for a very specific type of file format and metadata schema that also has very, very clear definition for every metadata entry. And we need everybody to have it in the same standardized format so we can actually integrate data, not only per organ, but eventually connect it across organ system, and also to be able to put it on different portals for queries, right? It needs to be in standardized format, so that needs to be done. That's for the matrices, so the summary data that doesn't have identifier. Now for the raw data, we appreciate that there's different challenges tied to raw data submission. Ideally, we like it to have in our HCA data repository, or at the very least for now, we'd like to know where the data was deposited. It's important for us to know where the raw data is, because we need to be, there might be future iteration of atlas. We may need to realign data. We may need to reprocess data as new computational methods are developed. And so ideally, it would be for every data set that is part of a version of an atlas. For example, immune right now is 14 or 15. Then we want to know where that data is, and ideally it would be all together as a collection. But for now, as we learn and iterate, we just need to know where it is. I hope this answers your question. Thank you.
Hi, Chloe. So it's an amazing presentation. I really enjoyed the live demo of the CAP.
Oh, great.
So I have two naive questions. So the first of all, seems like in the demo, like the interaction, the interactive with the website is very smooth, but it seems like you only show like probably hundreds of thousands of cells each data set. So do you imagine when it becomes like millions or 10 millions of cells, you still can do live computing at such fast speed? This is my first question.
Maybe let me answer because I'm jet lagged. So let me take one question at a time, and I'll take your next question so I don't forget. This is such a good question, and I appreciate that you understand the intricacies of this. We just spent a year and a half doing a lot of re-engineering to make this work, and so now we can work to a few million cells actually very smoothly. We also appreciate that there might be regional, we need to tailor our architecture so that regionally you can also access the data very smoothly. And so we've been developing regional architecture so that you can access the data very smoothly. And so we have a whole team of Python software engineers working on this. And that's one of our main priorities so that we can also, with time, offer services of automated annotation very rapidly.
Yeah, cool. So my second question is, I really like the voting for the cell annotation function. So what if I have a new cell subtype that I want to annotate? Is there a function that allows me to give new annotation to the existing atlas?
Absolutely. And it can be both in terms of your data set that you're annotating, or through, you're looking at, let's say, my integrated atlas, and you're like, Chloe, you missed a cell type. You actually can go and re-annotate my data set by creating a profile and propose a re-annotation and kind of give us all this information and your justification for it. And at the end, what we'll do, we'll open these annotation Jamboree, and depending on the size of the bionetwork, they might be fully virtual or hybrid. I can speak for the immune bionetwork, and hear from us this winter. It's going to be first virtual so that anybody can contribute to it. You'll be able to go through CAP, or you'll be able to reanalyze the data and provide your information back to GAP. We just ask people that they respect our embedding coordinates. And then we'll compile the information. We hope to have some initial results before the general meeting in Singapore. And so then in person, with access to a hybrid meeting, we'll be able to discuss where we disagree and agree. So for example, this will be an opportunity for you to say, hey, everybody, you forgot about this new cell type, and this is my justification, and for everybody else to kind of have a chance to have a discussion with you. So absolutely, that's an option.
Amazing, thank you.
My pleasure.
I think second row over here first.
I think.
Okay, someone else.
So I have two questions. So also the first one. So the marker genes annotation is always with on which cell against which cell or indifferent. Is it possible that to say, for example, I just want to see the T cell markers against B cells, not
Yes.
in general?
Absolutely. And I'm so glad you're asking all these questions. It means that it made sense to you. And because of sake of time, I couldn't go really deep into any of this. But when you try to define your markers, you can decide what's your reference. It could be the whole background, all the cells, or it could be against specific reference. So you can select what is your reference. What we ask when you provide the rationale for providing an annotation is you tell us. For example, we say, I define these marker genes based on this statistical framework, and I compare my CD4 to my CD8. Or I only looked at, for example, these T cells within CD8, and I tried to define what's a naive, what's an effector, so I did only within the CD8. So yes, you can decide on what's your background reference. Thank you and I think you had a second question.
Yes, and I want to ask how the cell type annotation that coordinates with this cell ontology. Is this related with this cell ontology annotation?
The location of where the tissue is from?
No, it's something like, gene ontology, there's also cell ontology.
That's right. I was referring to cell ontology here. And so for certain organs, the cell ontology schema is a little bit more detailed, but some others is completely lacking. So as part of the HCA effort and the cell annotation platform, we actually have people, including David Smith-Sutherland, that leads the cell ontology effort, and work with you to know how closely it relates to existing cell ontology term. And if it doesn't exist, you tell us it doesn't exist, and then we ask you what's the closest term so that we can work with the community to define a more precise term for every cell type.
There's one short last question.
So hi, Chloe. Really nice to see you again. So my question is, so this CAP system is really awesome. I just wonder, can we just find all the canonical markers provided by the authors? Because that's super important also for we apply it to some other data sets, right?
Absolutely. And so so thank you both for your question. So one of the fields we ask people to fill out is it's either can be marker genes have different categories, but we define them in two broad categories. Those that will be data driven, and those that can be they're legacy-based markers. So for example, if you ask me, you say, oh, Chloe, tell me about your favorite dendritic cells. And I say, oh, do you know about my axial cyclic 6 dendritic cells? These are my 2 favorite markers. You have a field where you can provide them with a rationale. And as we compiled our consensus framework in the cell card view, you're gonna get this information. And as well as it's conceivable that a few of us have identified the same cells but provided different markers, so we'll be able to compile this information, and with time that become the foundation of next iteration of automated annotation framework. Thank you.
All right.
Good?
Okay, fantastic to see all the excitement around CAP and the data ecosystem. It's going to be an incredibly valuable resource. Chloe, do you want to put out one call for what you want people in the community to do? Or,
please reach out. Even if you don't have a data set, do engage with the cell annotation platform team. When you get an email from the HCA executive office or from a different specific bio network, and it's one of the organ system you're excited about, do take part of the feedback mechanism, whether it's fully virtual, whether it's just going on the website and contributing to agreeing or disagreeing, or actually taking part in Cell Annotation Jamboree. We really look forward to engaging with all of you in the coming year. Thank you. Thank you all for your attention.
All right, thank you very much, Chloe. Okay, before you all head off for the break, just a quick announcement. Speakers in the next session, please come to the AV booth to check your slides. We're going to have a short refreshment break. Please come back at 11:10 a.m. after the break.
Hey, can everyone please take their seats? We're about to start the next session. It's going to be really exciting. It's on emerging technologies. My name's Alistair Forrest. I'm in Australia, and my co-chair is?
My name is Jiang Lan from CNCB, China International, China National Center of Bioinformation. Yeah.
Okay, and I'll leave it in my co-chair's capable hands to introduce the first speaker.
Yeah, okay. Please take seat, everyone. Let's go back to the session 2. It's my big honor to introduce our next speaker, Professor Huang Yanyi. He is a professor of analytical chemistry at Peking University, and also principal investigator in BioPeak. It's established by the top 2 university in China, Peking and Tsinghua University. And he is a pioneer and leader of several areas of cutting-edge technologies. He have being very successful to invent a series of technique and instrument, including sequencer and microfluidic device and spatial resolved imaging platform. Without any delay, let's welcome Professor Huang Yanyi.
Thank you, Lan, for a kind introduction, and I'm flattered that you call me a pioneer. I'm not sure I'm pioneer here. It's the first time for me to join the HCA meetings. It's a great honor, and thank you, Angela, for the invitation. I think today I'm just going to focus on something we are doing recently. It's the study, how we study this spatially resolved information. So I changed the title a little bit. I will discuss the technology development, the development we developed recently to illustrate the spatial information for RNA molecules, especially RNA molecules in the tissues. So let me see. Okay, so let me explain why I'm interested in this. My group has been working on pathology-associated genomics information for quite a few years. So we studied normal tissues as well as the lesions, the disease-related tissues. So that actually across, let me see, I don't see the laser pointer, but I think anyway. So it has been, well, anyway, I just mentioned this why. So the one of normal ways to observe these tissue sections under microscope, and you find something interesting in the tissues. Then you can locate to the tissue-specific locations, and you can cut it out. So this is called laser microdissection, right? And I'll give you a few examples. The first example is this. Like a few years ago, I collaborated with Professor Dongxin Lin and Chen Wu, and also my colleague Jianbing Wang at Peking Tsinghua University, and also Fan Bai at Peking University. So we have these 5 donors, the volunteer donors. When they passed away, they donated their bodies, right? So we can take out 10 of their organs, very small pieces of the tissue from the organs, and then we examine under microscopes. None of these people that die from cancer, so the other diseases, right, the aged diseases. And then we examine their pathology of their epithelium, all normal. So we take out the normal epithelium part, just taking the 200 microns from the section to section in between these sections, and another about 200-300 microns from piece to pieces. So every small cutting out of this micro dissector, cutting out the sample containing relatively 500 to 600 cells. That's it, and looks very normal and very uniform under microscope. And we take off these samples, we do the deep sequencing. And by doing so, you actually found very interesting information. You see the variation across tissues, across patients, and you do find a very interesting collective mutations in all these patients. Another extreme is you take out the cancer tissues. So a few years ago, we collaborated with the oral pathologist. We take out the oral cancer tissues, and there's a normal adjacent part of the tissue. Then we do the sequencing. Then we do simultaneous DNA sequencing and RNA sequencing. By doing so, you can find those gene fusions and copy number variations, abnormal copy number alterations. They can merge the data together to do the analysis. So the more we're doing this, the more we find that it's interesting. You probably need to observe something simultaneously, and you want to dig deeper and deeper. So a few years ago, I collaborated with a theorist. This wonderful theoretical computational biologist, or I would say computational chemist, Gao Yiqing, Professor Gao, actually working with us for quite a few years. We're doing all kinds of interesting machine learning problems. But particularly, we're interested to try to find the information flow between the DNA, RNA, and the protein. So we have developed an effective method called high-C2 geometry, CTG. This is a method we actually embrace the diffusion properties from physics and to try to understand this very sparse high-C data merged together and to visualize the interaction between the chromatins in certain part of the genome. So by doing so, you can actually find something very interesting. For example, one of the most important findings that the gene-gene interactions that revealed through this chromatin structure to geometry, which called CTG matrix, is highly correlated or can be used to predict the protein-protein interaction. So gene-gene interaction, GGI, it's a very powerful tool to predict PPI. So by doing so, you can imagine it actually guide us to try to think about how this cell, the molecules, bigger molecule inside the cell to regulate themselves, and also probably will actually review or unveil those possible drug targets by doing the gene-gene interaction prediction, right? And to better illustrate the importance and the chromatin structure in the tissue resolution, we'll apply the few mini-bulk multi-omics sequencing. So here's one. We collaborate with Yiqing again, and also Jianyun. That's an oral pathologist we have been collaborating with for about 10 years. So we studied two types of oral cancers, the adenoid cystic carcinoma, ACC, and also the squamous cell carcinoma, SCC. So this ACC and SCC, they look so different under a microscope, but we try to figure out why they're so different, and there's anything common in between, right? So we use laser microdissection to collect a few dozens of the small areas, small region of interest. So each one contains from a few dozen to a couple, say a couple thousand at the most, cells. And for this specific cutout, we use this sample to do different omics sequencing. You do RNA sequencing, we do high-C for chromatin structure, we do chromatin IP sequencing, we do RNA sequencing, we do DNA methylation, and all with very clear, defined pathological observations there. And we confirm that in oral cancers, okay, chromatin recognition is a critical hallmark that influence gene-gene interaction and transcriptional regulation. So in cancers, long-range interactions and the compartmentalization are clearly reduced. So it's very clear for both ACC and SCC. Also, the spatial chromatin features influence copy number alterations. So copy number alterations, CNA, are popular in OCC, but not so popular in ACC. It's quite rare, actually. So copy number gains and losses are closely associated with chromatin domain boundaries. And this has been observed in many of our and other studies before, right? But more interestingly, is that these boundaries not only present in the samples with copy number alterations, but also in those samples with normal copy number profiles. The samples without copy alterations, you also see those closely compartmentalization boundaries there. So this is indicating a structured predisposition to genomic alterations. It's very interesting. And also, in the 3D chromatin structures, it seems very stable. So more fundamental than histone modifications and DNA methylations. So it seems like it's more groundedly built markers for us to discover more hidden geometry related methods in cancers and also in other diseases. So due to time limit, I will not cover more details. It has been published. It's quite a long story. We measured quite a lot DNA sequencing data there. So in cancers, methylation change is actually happening in very early stages if the methylation will change. Why I say this is because in ACC, you do see the methylation pattern looks very similar to the normal tissue. But in SCC, they change quite a lot. In open C region, you see the hypo or demethylated patterns are everywhere, right? But if you see the CTG matrix or all the chromatin structures as the input using the multi-head attention machine learning model, so you can achieve a pretty good prediction power even for other diseases. So you learn from ACC, oral ACC, those signature can actually apply to other adenocarcinoma. It's very interesting. So we start to explore more and more about this. So why I say this? By doing this, I felt very frustrating. So the more data you gain, the more questions will actually appear, right? So you thought you probably would do the single-cell sequencing more and more, then you can answer this question, but it turns out it's just give you more questions. So I had these two approaches. One approach is I try to build a sequencer, Jianglan just mentioned, like as accurate as possible. So it's not main topic today, so I just mentioned a little bit. So is this sequencing accuracy now enough for you to do all these experiments? Probably not, right? So recently, we continuously elevate the sequencing accuracy from Q40 to Q45, and now we are reaching Q50 in the past few weeks. When the new rounds shows we can actually hitting the Q50 data. So once you're hitting the Q50 data, now you observe so many artifacts generated by the sample prep and sample storage and whatever you've done in this library prep sequences. Okay, and the second part is like, I never really thought what I did before, like a microdissector sequencing can be described as a spatial omics. But Leo Patrick, they wrote this paper, it's an interesting title called the Museum. That means probably all this technology will be obsolete quite soon, right? So in this museum, interestingly, they put a laser microdissector is one of the methods, a quite important method to do the spatial omics. So I'm glad now we are in the same page, we are thinking about the spatial omics. So I look around, okay, now you have so many commercial available companies can produce these wonderful machines to do spatial imaging-based sequencing, to do capture-based sequencing. Okay, I'll try to focus on something probably not that popular. We'll try to do developing the in-situ detection methods because it's not that popular in commercial available machines. So, okay, I'll present two of our approaches. Okay. One has been published, I'll go very quickly, called Spring-Seq. So this approach is based on my personal training background. So I have been working on microfluidics for quite a few years. So you probably will listen to next speaker's talk. They also will do this. So we know each other for more than 10 years. We're working on this same field. Okay. So this is probably the simplest ever microfluidic device I made in my group. It's a double-sided tape. Cut a channel of this tape, and you glue two glass slides together. And in one of the glass slides, you drill two holes. Because why two holes? You need a liquid in and out, flow through it, right? And then you put your slice, your sample, just in that channel, so they can bring the reagents in and out, and you can change it. So I made this instrument in the lab with all these wonderful students in the pandemic time. So you can just squeeze in the lab to assemble this. And it's quite simple. And using this simple instrument, we did in-situ sequencing. So think about this. The flow cell there, it's a sequencing flow cell, sequencer flow cell. So we borrowed Illumina sequencing reagent. We just pump in to do the sequencing. But what to sequence? We use the PETA probe to capture those genes you want to study and turn this gene into a barcode, DNA barcode. So you just sequence the DNA barcode, you actually get two important information, which barcode is there and where it is. So the barcode represents the gene type or isoform type, I will mention it a little bit. But the location is actually recorded just by taking the image. So now you have exact in-situ information of the gene, right? So let me show you just one example. This is a corona section of the mouse brain. If you use a 40x objective, it takes about 500 pictures to cover this whole brain, right? So, and then you need to take 10 cycles, we calculate, to capture this whole brain, to 20 images, with 2 color channel. And with DAPI, 21 images. And you take 9 different Z sectioning for the focal adjustment. So 9 times 21 times 500. So turns out it's about 100,000 images, 1 terabyte, all finished within 10 hours. And the sample prep within 10 hours. So that means within a day, you get the data from the fresh tissue. You slice it, next day you get this data. And then all this data, they give you over 10 million dots if you take 100 genes for study. Then you can actually use these 100 genes. Then you can annotate the cell type, and then you can project back. So you get this image to illustrate where is which cell. More interestingly, now you have this information that cannot easily be reviewed by other technology if it's not in situ detection. Now you know where the gene located inside the cell. So is it located there, like distributed evenly, or is it polarized distributed? By doing so, you can actually gain a lot of interesting information. It has been published, so I've skipped this result and just give you those summarized. It's a very fast method. You can get from tissue to the information within a day, right? It's very sensitive, capture about 30–40% of the genes by sensitivity per se. You just take the measurement, it's about 30–40% capture rate. And it solved the crowdedness issues by some tricks. We mentioned in the paper, and also we built a new coding scheme called the hybrid block coding. It can scale up to much bigger tissues very easily, and using this, you can study some animal models I didn't mention here. Okay, so and then I showed this to Angela a few years ago. So Angela challenged me, say, can you do something more interesting, like can you detect the isoform? We tested for Angela, and I actually don't know what isoforms she's interested, but anyway, we are a service provider. We can do this, right? Okay. Now it turns out a question. Angela is also expert of the microfluidics. Why didn't she do this by herself? Right? Why a microfluid developer, when they develop microfluid devices, they were stuck on this device to do service for everybody? I hate this. But more importantly, my student hates this, so they stop working on many of my colleagues. Okay, I think, okay, probably we need to set back. Why is this? Because it requires cyclic reactions. They require specific engineering tools. You need to control the liquid in and out. It's not that trivial for most biological students, right? They just get into the lab and they need to learn how to program this, that. They're busy enough, they don't want to do this. So, and is this the only way you can do this information interaction? Not really, right? How about we do a single run reaction? That means you can do this pipetting on any bench in the world. And then you can take an image, use any microscope in the world, in your next door lab, right? You take it, and you do the simulation, you do the computation if it's needed. And people have been doing this for a long time. They try to increase the plasticity for imaging by using all kinds of methods. But many modalities are not common. So you either to use very special microscopes, or you need to do this over and over again. Long time, very tedious, right? So with collaboration with Professor Chun Hongzhen at the PKU Medical School, we come out with this idea called PRISM. How we can do this multiplexing, super multiplexing, for example, 30 to 60 plex imaging, fish imaging, like within a day, using all normal equipment and a normal microscope anybody can access. Right? So it's a very simple idea. You just need to tune the barcode into different sections, and for every section, you actually can code it with a different color and the intensity. So it's really the color coding. But by doing this, there's one trick. You probably need to mix the ratio of the color, the dye-labeled probe and the unlabeled probe with a specific ratio you already noticed. Then you can just go through it. You just let them sit in there to stain by itself. And there's one interesting question. Because this color barcoding space is huge, right? But it will actually mix with one particular question. Is like if you amplify this nano ball to small ball or to bigger ball, that will affect the coding space. So it's actually turned out that we figured out it's a need to be vector differentiable coding that reduce the color space, but still enough for you to do many plex sequencing. So I show you a few examples. That's the 30 plex imaging for the mouse brain, just using the normal 4-color microscope. And you can use the 30 plex for mouse development as well. You can use this to study all these detailed developmental stages and how the organ formed in different days of the mouse embryos. You can do this within a very short period of time. This has been done within a week. Just take a few tens of slices, do it very quickly. Right? And we do the 20 continuous sections for the liver cancer. This has been done within 4 days. You just stain it, you just image, and you turn out you have more than 1 million cells annotated. You can see how these cancer-associated fibroblasts block the invasion of the T cells, they cannot really get into the cancer. And you can also see the different exhaustion patterns from the liver, normal liver part highly affected by the high HBV, and also other part is the tumor part, but they just cannot really access to the active T cells. Yeah. You can also see the structure using the CRC samples, right? Colon cancer samples, you can see this crypt of the intestine very clearly. You can do 3-dimensional. This is a mouse brain. It's just 3-dimensional using the 100 to 200 micron thick slice. You can see through it. I will skip this because of the incompatibility of the computer, so I skipped the video. Otherwise, you can see, you can go through it 3-dimensionally. You can extend it to 60 plaques as well to do mouse brain. To do that to 3-dimensional with 60 plaques using light sheet microscope, scan very quickly the big slice. You can go through very quickly, see how this 3-dimensional structure organized, and you can annotate the cells 3-dimensional in situ. And with that, I will thank you and thank all my students and collaborators. Thank you.
Fascinating methods, Yanyi. For PRISM, I guess the obvious application is in some clinical diagnostics or something. Is that fair to say? And so then, I didn't catch that, is it FFPE compatible?
Yes.
Okay.
We actually tested it's very fully compatible with FFPE slices. So that way you can use the archives quite easily. Yes.
Fantastic.
Sure.
Hi, everyone. I've just got a question from a virtual participant. Thanks for the excellent talk. I would like to know if the subcellular data mentioned is open sourced or not.
Sorry, I thought. Yes, it's fully open sourced. We actually built a small website. I didn't put it in my bag. Yes, you can put a data set and website. You can play with the data. It's fully open source. Yeah.
Thank you.
Sure.
So I'm wondering how the data looks like in the UMAP space. Did you see any kind of cross-contaminations of different cell types?
It also depends how you choose those marker genes. So we actually get this information using the single cell data from the public domain. You need to analyze and pick the best set for the particular question you try to study. Yes. Yeah.
Okay, can we please thank Yanyi? It was awesome. Okay, so now I'm proud to introduce Hirofumi Shintaku. He's a principal investigator at RIKEN, and he's also a professor at Kyoto University, and we're really excited to hear you speak today. Thank you.
Thank you. Thank you, kind of introduction. All right, so this is actually the first time, even for me, attending a HCA in person, so I'm very excited to be here today. So I'm going to talk about nano-electrokinetic transcriptomics to link the gene expression to single cell behavior. Right, so before I forget to mention, I like to show this slide. I like to acknowledge all our team members and the students at RIKEN at Kyoto University for their contribution to the projects which I'm going to talk today. So our group, right, so this is one. So our group utilized micro-nano-electrokinetics to expand a single cell mix. So this movie is showing one example how we utilize nanoscale electrokinetics. So we here use nanoscale focused electric field to do something interesting. So we actually apply this nanoscale. Oh, sorry, this doesn't work. Okay. Apply nanoscale electric field to permeabilize the cellular membrane without killing the cell, and we deliver molecule or extract molecule from the cell without killing it. So we here show today the 3 examples in which we utilize the micro-nano-electrokinetics to expand the single-cell mix approaches in terms of resolution, temporal, and phenotyping. So as you know, so single-cell RNA sequence is very powerful, but yet it is still endpoint measurement. So we basically kill the cell of interest upon the measurement of the RNA. So our group has, so many people have put a lot of effort to develop a computational approach to infer the dynamics of single cell, including the pseudotime, RNA velocity, but still these are endpoint measurement. So our group has proposed a unique approach so-called SYNC-6 that leverages the microscale electrokinetics. So I want to start with this, our idea. So our basic idea is we try to see the gene expression difference in the nucleus and cytoplasm. So we basically think like nuclear RNA expression may contain the future information, and cytoplasmic RNA expression contain present information. So by taking those 2 RNA species difference, we can probably see the dynamics. To profile those 2 RNA species in the same single cell, we have developed a microflex system that physically fractionates cytoplasm and nucleus from the same single cell. And with this approach, we can quantitatively profile the gene expression in cytoplasm and nucleus, and also we can, employing the long read sequencing approach, we can even study the structure difference and diversity of isoform in nucleus and cytoplasm. But as you have seen here, these are still endpoint measurement, and we actually wanted to develop a truly dynamic gene expression analysis without killing cell. So to tackle this, we leverage nanometer scale electrokinetics. So what do we do here is we basically use a glass capillary, a conventional, glass capillary within nanometer scale in a diameter, and we gently touch the surface of the plasma membrane and apply pulsed electric field and leverage the nanometer scale focused electric field gently permeabilize the cellular membrane and extract the molecule. So this method is really gentle. So we apply this to C. elegans embryo, and we call this approach as live organismal transcriptomics. So we basically track the gene expression as time series with observing the embryogenesis and monitor until hatching. And here we apply our nano-electrokinetic sampling at single to 4 cell stages. So this movie is showing how we do our extraction. So the shadow on the right side of this movie is a glass capillary, and we have a nanometer scale. This glass capillary has a nanometer scale in the diameter. And by applying pulsed electric field, we can gently permeabilize the cell membrane, and we sample mRNA in the order of picogram. And the cell divides into 2, and then we sample, and then we wait for some time, and again we sample again from those 2 cells. So actually the embryo sitting next to the sample one is a control, so we didn't do any sampling, and we keep on observing their behavior after sampling. And after sampling, the embryo keep on dividing, and we can see they're hatching quite normally. And so using this approach, we can obtain very unique data set like a gene expression time series, and we can also obtain the microscopical observation of the live imaging. And combining those 2 interesting data set, we can identify the important gene that defines the embryonic weight. Right, so now I want to move on to the last topic. So elastomix. So we use nanometer scale electrokinetics for expanding the phenotyping spectra. So this method is called elastomix, electroporation-based repeat bilayer assay for cell surface tension and transcriptomics. So we here use nano-electroporation to profile the mechanical phenotype. So mechanophenotype is an essential cellular characteristics. It is involved in many biological contexts. Cell division, obviously, which is the most fundamental process, involves the regulation of the mechanical property of the cell membrane. And other example can be found in cell migration or tissue development and so on. However, at this moment, there are no single cell approaches to bring such a mechanical phenotype to the omics layer of the single cell at high throughput. So this situation motivated us to develop a method to profile simultaneously mechanical phenotype and omics layer of the single cell. And here we forecast the cell surface tension, which is one of the important mechanical property of the cell. The cell surface tension is approximately sum of the plasma membrane tension and the cortical tension. To probe the cell surface tension, it's counting down. Yes. To probe the cell surface tension, we focus on the change of the free energy before and after the pore formation in the lipid bilayer. The change of the free energy can be described by this equation. So if you look at this equation, you would notice that this equation contains the parameters, surface tension, and also radius of the pore. So this equation tells us that the pore formation is dependent on the cell surface tension, and the radius of the pore is dependent of the cell surface tension. So this means that we can profile the cell surface tension by measuring the radius of the pore. Based on this idea, we developed the elastomix. So next slide. This slide shows how elastomix works. So we first see the cells on the surface of the tract membrane. So tract membrane is a non-conductive film with a bunch of nanopores. On the SEM image, you can see the cells are growing on the surface of the tract membrane. So you can also probably see the tiny pores. So these pores are through holes in the non-conductive film. So the cell can grow happily, and also we can apply electric field across this nanopore. So in the center image, as you see here, we place two planar electrodes across the tract membrane, and we apply pulse electric field again. In the meantime, we deliver molecule called DNA tagged dextran. So this is actually the dextran molecule tagged with the DNA. And we prepare this DTD with various molecular weight, and we deliver the DTD into the cell. And as I mentioned, the radius of the pore is dependent on the cell surface tension. So when the cell has a relatively low cell surface tension, so they create a smaller size of pore. So under that condition, we can deliver only the smaller size molecule, only smaller size DTD into the cell, and less amount of DTD. The cells with high cell surface tension creates larger size pore. Under that situation, we can deliver more DTD or larger size of DTD. So just by profiling the amount of DTD or the size of the DTD, we can tell the cell surface tension of the cell. So after doing this nano-electroporation, delivering DTD into the cell, we detach the cells from the tract membrane, and we transfer those cells into just standard 10x genomics or single cell mRNA assay, and we profile the transcriptomics as well as the DTD amount. So we can know the gene expression from the transcriptomic data, and we can know the cell surface tension from the expression of DTD. So with this elastomix approach, we can explore the diversity of the cell surface tension among different cell types. So here we assayed cancer cell lines with different malignancy. The highly malignant cancer cell tend to have low cell surface tension. So here, for example, PC3, MDA-MB231, a highly malignant cell, they tend to have low cell surface tension. Therefore, the count of DTD is lesser than the MCF10A or MCF7. And we apply this approach to a bunch of different applications. Here today, I'd like to share with you one example where we apply elastomeric to cellular senescence. So cell aging due to the replication causes increase in the cell surface tension. So this increase in the cell surface tension may be, maybe involved in the age-related diseases, but a gene regulatory mechanism behind the change in the cell surface tension and its cellular heterogeneity remain unexplored. To explore the cell surface tension around the cellular senescence, we here applied elastomerics to TIG1 cell, which shows the replicative senescence. So TIG1 cell increase their cell surface tension around the cell replication. So the pictures on top are showing the fluorescence images of the TIG1 cell after nano-electroporation. So green colors are showing the amount of molecule delivered by elastomerics. As you can see here, the TYIG1 cell PDL66, which is relatively aged one, gets more DTD. So we can profile the cell surface tension increase around the cellular senescence, as you see here. And we can also, by doing the 10x genomics, we can also quantify the increase of the cell surface tension by counting the DTD. And combining the cell surface tension information and gene expression, we can study the interplay between the cell surface tension and the gene regulatory mechanism. So here we study, we show, sorry, I'm not good at playing with this. So on the left, we are showing the correlation between the cell surface tension and the gene expression. So as you can see here, we observe positively and negatively correlated genes, and we actually observe genes tend to upregulated around the cellular senescence shows a positive correlation, and downregulated ones show negative correlation. And to identify the regulatory gene to the cell surface tension around the cellular senescence, we here applied multiple regression analysis, and we identified RRAD that shows a high contribution to the cell surface tension around the cellular senescence. So RRAD is known as a tumor repressor, which represses the glycolysis mainly through the inhibition of GLUT1 translocation to inhibit the import of the glucose. To confirm our observation with our approach, so we first applied a siRNA assay to RRAD to reduce, to downregulate the RRAD expression, and we actually observed the downregulation of the cell surface tension regardless of the cellular senescence level. And the second, we inhibited the glycolysis by 2-deoxyglucose. That increased the cell surface tension in TIG1 cells. So these results indicate that RRAD is a key regulator of the cell surface tension along the cellular senescence in the glycolysis axis. Right, so today I introduced 3 examples of our approach leveraging micro-nano-electrokinetics to expand the single-cell omics. I hope you could enjoy one of those. And with that, I'd like to close my talk and take any question from the floor.
Okay, I've got a question straight away. That was awesome. I was just wondering about the cell surface tension during cell cycle. Have you ever looked at that during before and after mitosis?
Before, we actually didn't see the cell surface tension dependence on the cell cycle.
Okay.
Well, we actually wanted to see that and expect to observe that, but I think Calvin sensitivity is not high enough to detect that, I believe.
Oh, you?
Thank you. So I saw you have 0.1 picogram RNA. So what is the percentage of this to the total RNA? So let's say you have 1 million copy of RNAs, and how many RNAs you get for each, how do you call that, the stick?
Yeah, so, well, in case of HeLa cell, for example, we take about 1 picogram, and HeLa cell is, our HeLa cell had about 30 picograms, so it's like 3% of the total.
Okay, thank you.
We got one at the back.
It was a great talk. I wonder if you plan to perform perturb-seq on your surface tension technique.
Yeah, that would be a great idea. So we hope to try that, but we have not tested that yet. Yeah. So just switching, the polarity of electric field, we can deliver or extract. So we can introduce some functional molecule, and then we can study the cell surface tension.
Do we have any more questions? Oh, yeah, we actually got one at the back and one at the front.
Hi, thank you so much. It was really a nice talk. I was just wondering about the nanoscale temporal sequencing, which you were talking about for the live organismal transcriptomics. So if we try to apply in order to see how a cell develops, as you have mentioned that we need to have a control cell. So how can we do that when we want to look at a single cell development?
So I cannot find you. Where are you? Oh, over there. So your question is how we control the amount of the RNA, for example?
No, if we try to do a single cell, we want to look at the development of a single cell using live organismal transcriptomics. What would be the control cell? How can we look at the control?
Okay, so in our case, so we had the same strain, and we know how much success rate for the hatching, and then we check if that success in the hatching rate doesn't change, even with the sampling, and we make sure. So in our particular case, so actually I didn't explain. Oh, this doesn't move. I don't know. So in our particular experiment, we tested OMA1 mutant that shows 50% of lethality in the embryogenesis. And when we tested that, we check if the success rate in the hatching change or not, and we control how much amount of messenger RNA we extract. And once we make sure that the amount is safe enough, we ask our biological question. So we need to tune the invasiveness depending on the question we want to ask. Does that answer to your question?
Yeah, thank you.
Very nice talk. I just wonder whether you have a way to measure for the last part of the work, when the cell incorporate the DTD, whether they have some expression change induced by this incorporation.
Incorporation.
Whether this DTD can induce some gene expression changes.
Oh, I see. So we of course tested whether that influenced the gene expression or not, and we always test how much DTD we deliver, and we try to minimize, and we have to balance between the sensitivity and the invasiveness. So we change the Well, I'm not sure I'm answering to your question.
Thank you.
Okay, in the interest of time, I think we need to move on to the next speaker. I'd really like to thank you, Hirofumi. And if you got any questions for him, please see him after lunchtime. Thank you. Okay, and our last speaker for this session is Professor Gang Chao from Shenzhen Institute of Advanced Technology. Go ahead, thank you.
Okay, thanks for the introduction, also the invitation from the commit. So I'm Gang from Shenzhen. So as we are overdue already, I will go quick, and hopefully you can get lunch earlier. So today I'm going to talk about the integrating of single-cell spatial-omics and Connectome. So as we know that, so to deciphering the connectome of our brain is a central question of neuroscience, and it's on top of that, it's also pretty important for the artificial intelligence, which holds the potential to revolutionize the machine learning. So, but still, it remains very challenging to decipher this connectome at a spatial level, because it's already very hard to decipher the spatial transcriptome and also spatial proteomics. So that's why, as we know that, although all the spatial-omics looks very fancy, so when you look at the data by yourself, you will see that there's still a lot of problem. Although there are a lot of methods developed by different labs, there's still a lot of problems of this spatial-omics. So they are all very exciting, but it's still at the beginning stage of the spatial-omics. So we are also trying to develop and improve this spatial-omics. So we spent 9 years to develop this MIPSIC, in short for multi-omics in-situ pairwise in-situ sequencing. So since this one has been published in this year, I will not go to the detail of this method. But I just want to show you that when we combine the one challenging of spatial-omics, it's very hard to reach the single-cell level. So we try to stand the membrane with some region so that it can we can visualize the membrane. In this way, we can more or less say the single-cell level of this expression pattern of the different genes. So because we're using in-situ sequencing, we can also see the SMP from different genes. So with that, we can see the allele-specific gene expression of each gene at different position. We can say at which cell, in which position they express the gene from their pap or from their mom. So actually, it's quite surprising to us. It's very in the beginning, we thought that probably it's very even, but actually there is very unbalanced. And we are also trying to do the 3D spatial-omics by at the whole leg level, but it's still very challenging for the imaging. So that we start with some zebrafish brain, but as a proof of concept, we can actually reach the 3D level for this spatial-omics. And as I mentioned, we are also want to do the spatial proteomics and other multi-omics for DNA and RNA, and sometimes we also want to check the neurotransmitter, so that we combine the antibody conjugated with DNA. With that, we can transfer the the HCA to in-situ sequencing. So that with that we can simultaneously detect DNA, RNA, and protein. And sometimes for the neurotransmitters, they also have antibody, for example, GABA. So with that, we can do the multi-omics. Here, we are simultaneously detecting 30 proteins and 6 RNAs for the cancer sample. So with this, we can see the spatial map of the cells and the cell-cell interaction, and also the panel, the expression pattern of each genes. Okay, and we can also combine the spatial-omics with other imaging technique, because we are working on the imaging-based spatial-omics. So we can combine our spatial-omics with custom imaging. So first, we perform in vivo custom imaging firstly, and then we try to we deliver a dye over there, and then we find back the dye, and then we perform the spatial-omics on the same region, so that we can combine the custom activity recording with our spatial transcriptomics. So with that, we can say, okay, for example, as you can see here, the excitatory neuron, they have a significant higher custom amplitude, while the inhibitor neurons, their frequency is higher. We can also combine with other imaging techniques, such as Raman imaging, as we show here. Okay, so this is a part is about the spatial-omics. So I will spend a few minutes to talk about our recent work to combine the spatial-omics with Connectome technique, which we call spider-seq. Okay, so the basic idea is that, so if we want to trace in our Connectome or neural network, we use a virus, neurotropic virus, such as rabies virus. So this rabies, people using this to trace in the virus in the beginning with GFP or other fluorescence proteins, but unfortunately there are very limited fluorescence proteins. You can trace in only a couple of neural circuits. So we replace this fluorescence protein with a DNA barcode, so that this neurotropic virus will carry the DNA barcode to the upstream or downstream neural circuit. And then we perform in-situ sequencing or spatial-omics so that we can sequence the DNA barcode at the in-situ level. With that, we can decipher the Connectome. So we actually we are doing a lot of engineering of this neurotropic virus and develop a lot of neural circuit tracer. And now we are using this DNA barcode to replace this fluorescence proteins. And then, okay, I think something wrong with this. Okay, we develop a technique called spider-seq, in short, for spatial-omics and okay, whatever. So this is a new technique called spider-seq, which we incorporate a DNA barcode in the neurotropic virus. We call it a spider virus. So this spider virus will carry the DNA barcode, and we inject them into a different region of the brain, and then this spider virus will retrogradely traffic it to the middle prefrontal cortex region. And then we will dissect this middle prefrontal cortex for single-cell sequencing, for in-situ sequencing, or spatial-omics. And we, by the information for the DNA barcode, we can also get the information for the Connectome. So with that, we can combine the Connectome, transcriptome, and spatial-omics in one mouse brain. So we spend a lot of time to optimize the single-cell sequencing for the brain, because it's very hard to get the high quality of neurons single-cell sequencing. After 1 or 2 years optimization, we can get around 5,000 genes per neuron. So with that, we can get a lot of low-expression gene profiles. For example, the receptors and for the neurotransmitters. So within this single-cell sequencing profile, we can also say the DNA barcode, which represent the Connectome informations. And then we go for the spatial-omics, because we are doing target spatial-omics. We don't do the unbiased capture. So with that, it's much cheaper. Also, it's very robust. And so we can do a lot of slides, and then we have a very high dense slides for the middle prefrontal cortex. With that, we can reconstruct the 3D transcriptome in for middle prefrontal cortex, and we can also see the projectome information based on the barcode information. As you can see here, each cell cluster of each transcriptome cell cluster are shown on the left, and on the right you will see each projection clusters. We can also say that the configuration of those cell population are different. Okay, so as I mentioned in the beginning, when you see this spatial-omics, the data always looks very fancy. Very beautiful, but nobody knows if it's true or not. So we developed a fish technique called PyFish, and then this to validate this information. So as Yanyi said, a lot of people asking for help with this. So the students are too busy to provide this resource. So eventually we said, okay, why not just commercialize this RNA kit? So we have this kit available for the RNA fish. So with that, we can confirm all the gene expression pattern, as I showed in the beginning in this video. So that we also perform other controls with different repeat, and also we compare our data with other people's data for the Connectome data. Then we actually it's all good looks. And then with this we have the Connectome information. We have also the gene expression pattern. And for example, the neurotransmitter receptors, so that we can see the information flow in prefrontal cortex. So, and we can also see that the neurotransmitter released to reach downstream target. As you can see here, each circuit express different neurotransmitter receptors and also neurotransmitters. So, and as you can see here, also the neuropeptide are distinctly expressed in different circuit. So we mapped the information flow in the middle prefrontal cortex. And interestingly, we also found that different circuit express different kind of neurovarying molecules, such as the neuroligin, neuroxin, and also the axon guidance genes. And so that means probably we can use a gene profile to predict the gene circuit information. But in the beginning, it doesn't work well because we ignore the spatial information. And later on, we incorporate the spatial information together with the gene profile, and we can precisely predict the gene's circuit. We also test that in other data set to validate the generalization capability of our method. So we just downloaded the merfish data, and we can use the merfish data to predict each cell's the circular information. And actually it looks it's pretty precise, and we are still working on that. And then so I think I'm running out of time. So thank you very much. And also I get a lot of help from the students and people. Okay, thank you.
Thank you very much.
Yeah.
Okay, so we've got some questions.
Okay. Okay.
So what is it? I noticed PySeq?
PyFish.
PyFish.
Yeah. So it's a fish technique. Yeah.
So what's the advantage of that over the Spider-seq method? What's the contrast between those two methods?
Okay, the PyFish technique is it's a fish technique. It only allow us to do maybe several genes, maybe 9 or 10 genes. Okay. While the MIP-seq is a high throughput in-situ sequence method. It can go a few hundred or more than that.
Okay.
Yeah.
We got any more questions? Any from the online audience? Oh yeah, we've got one here in the middle.
Okay.
Thank you, wonderful talk. So I'm wondering how many types of DNA, RNA, and protein you can detect simultaneously?
We can detect DNA, RNA, protein simultaneously. You mean types? Let's say we can also detect long-coding RNA, like long-coding RNA, or we also tried microRNA for this kind of a short RNA. Yeah.
Okay.
Yeah.
I see.
Yeah.
Yeah.
Okay.
Okay. I think in the interest of time, we might wrap up. First of all, I'd just like to thank you to all speakers. It was awesome. And to my co-chair, of course. Do we have any announcements?
All right, awesome. Wonderful morning session and great participation. I think a lot of good questions as well.